Well, hello again from 2.5 kilometers below the ocean. We're coming to you live with footage from the remotely operated vehicle Sebastian. Broadcasting live from research vessel Falcor 2. And we are back on the bottom on the East Pacific Rise. Returning to the area we were at yesterday. Yesterday's dive was primarily intended to locate a potential site for experiment number 3. And we did that. We located a site. We left a frame behind. Now at the beginning of this dive, we are going to check on experiments 1 and 2. And then we are going to move on to the deployment of experiment number 3. Again, these mesh boxes are the experiments. And these are sealed down to the floor, so we're just kind of checking on them. And then we'll move forward. You can see the frame on the very right side of your screen there. Just leaving view now. And that frame is where we'll be placing experiment number 3. This will be the last of the mesh box experiments, but those are going to stay in place for most of the rest of the cruise, and then we will be collecting those back up. See here, just looking at the seal that's at the bottom there. It looks like a bit of it has sort of oozed out from under the box there. And this is the Underworld of hydrothermal vents is this cruise. So what we're looking at, or what we're looking for, is evidence of the venting fluids of this hydrothermally active area. These fluids are driven or pulled down into the ground. They are then heated and driven upwards back out of the ground, and that's where you get these hydrothermal vents, is where that fluid is leaving. But there's an interest here to see if those venting fluids bring along with them some of the fauna that are found at these sites. So these boxes are intended to provide a way for us to check for that. They're placed down on top of the vents, and then they are kind of left there for a period of time, but there's a stain that's been applied on the inside. So everything that was inside the box when it was first placed should now have a stain to it, a color stain. And when we collect the box back up, when we collect the fluid from inside the box, and when we sample the substrate below the box, if there's anything there that doesn't have that stain, that could be an indication that it arrived via the venting fluids underneath the box. Folks are starting to trickle on in, so welcome. Good morning, or afternoon, or evening, wherever you might be joining us from. We usually see our audience kind of shift around the globe throughout these dives, so welcome. We'll keep doing a recap here for the first hour or so, just as people trickle in. But we are back at the East Pacific Rise. Just checking on our first 2 experiments before we move on to the placement of experiment number 3. you can see a little bit of that silicone caulking down at the bottom there. So there are lots of little cracks in the seabed here, and that's what a lot of the venting comes up through. But where those cracks cross the footprint of the box, they want to seal that up. So we used basically what we would use to repair the bottom of a swimming pool, just an off-the-shelf sort of silicone caulking that we actually spoke about this. There was a lot of testing going on to figure out which one would work the best for this sort of application in salt water, in trying to seal up against the seafloor. So this is experiment site 1. This was the first one that we placed, which we weren't really sure how any of that placement would go. This was something that hadn't been done like this before. It went pretty well. And then experiment number 2, just continuing on with our trend. Today we'll have experiment number 3. You can see there some of that silicone that's been pressed out a bit, but it looks like there's still part of the seal underneath there. So right now, controlling the camera and over in the side seat, we have Sabine. And then our pilots for this morning, we have Chill as pilot and Alex as co-pilot. And then I'm also named Alex, and I'm over in the marine technician, or in my case, multimedia technician seat. Just making sure our stream is getting out there. We'll do our little weather check here, speaking of getting the stream out. Looking a little overcast out there, so hopefully we don't see too much interference on the stream, but if we do, if you buffer a bit, just have patience with us, please, as we work to bring the deep sea to you. Yeah, the silicone seal along the bottom of the box is quite soft. So when we're putting these boxes down, it tries to just kind of go down and be done. It's good because it allows them to really press that box down against the seafloor, and there's weights around the inside of the box as well that hold it down. So it should conform to all the little grooves and valleys, aside from the more substantial cracks, which is why we need the pool caulking. But at the same time, you can see kind of where it's smushed out a bit on experiment number 2. But it looks like there's still a barrier of the silicone underneath the edge of the box. So this metal frame in front of us, if you weren't with us yesterday, this is going to be the site for experiment number 3. That metal frame sort of marks out the footprint of that experiment. And it also provides an indication of where we may need to apply the silicone sealant there. As you can see, for example, the side facing us, there's a little bit of a gap there on the like right half of that. In between perhaps the second and third of this side from left to right. If you're joining us on YouTube or Facebook watching this live, feel free to type any questions you might have or anything related to that in the chat, and we will try to answer those as we can. If you've seen experiments 1 and 2 deployed, we do get a little quiet in here. It is a tenser situation, kind of getting those down as nicely as possible, but we will try to get those answered as we can.
I think that's it, so it's like 1, 2, 3, 4 places. Okay, and I think that here you made, oh sorry.
Go ahead.
That here I think there's some little, you made a little, I don't know who made it, but like
Yeah.
a little mark
Yeah,
here.
mark
and
there, mark there.
Yeah.
So here we have some of the fauna of the area. Of course, the large Riftia tube worms with that red lipstick appearance, as well as the sort of golden brown mussels in the area. There was a little white vent crab down in between those worms there on the clump.
Yeah, cable for that thing over it for sure. So just in case, I'll be ready to grab that from you and get a reposition.
Yeah, so the objective of today's dive, primary objective, is to get this experiment deployed. After that, time allowing, we'll be traveling back over south where we are now to where some student experiments were deployed yesterday, along with a little art installation from our artist at sea. The student experiments were various types of food on a tray, and also there were some beans looking at how they would be impacted by both a venting area and a non-venting area of the deep sea. A little bit of a tangle here, so the pilots are just working to get our stuff more nicely stashed.
Just got a power left or something. Get the tip over.
So that instrument there, if you haven't been around for us using that, that is our high temp probe. Lets us kind of probe the temperature of the venting fluids here. And the instrument that's being held in the left or port side manipulator there is our suction sampler tube. Think of it sort of as a large undersea vacuum cleaner. With that, we can sort of suck up some of the looser and smaller items here into a collection tube, a collection canister on the side.
Yeah, as long as you get it started, we can flip that.
So there is a little sort of sheath off to the side for that temperature probe. With the experiment in place, it's a bit difficult to see it, but if I switch over to our situation camera, you can see it just barely sticking out the front there on the right side or starboard.
Let's just move on. We're going to say we're messing this for too long.
Sample jar 1 to open.
Okay.
Sample 50, no?
Mm-hmm.
Finer.
Yes.
50, 15,
We're in the water, sure.
Jar number 1. Jar 1.
Like pull you in a little bit.
And if you could watch out that the rock is not splitting on the surface.
This one, so 3, so
1.
1.
Make sure the rock is not splitting on the surface. You said to make sure the rock is not
Yes, so
splitting.
if you touch like with the suction sample on
Couple
the rock,
questions here, I see.
then it's very difficult to look at the
First off, a question about what the beans were in the students' experiments. So the student experiments took 2 primary forms. One was trays with different foods sort of attached down to them. And those foods were like liver, meat, fat, starch in the form of potato. There was some other vegetative matter there I can't recall off the top of my head. And just sort of seeing what the vent creatures here would go for. And then the other experiment was the beans, sort of a mix of beans, including the mung bean, and those were left in both a venting site as well as a non-venting site, and just kind of taking a look at how that would impact those beans. Another question here of what one would study before marine biology, and just getting grips in biology is kind of the path that leads there, more through like traditional means. biology, chemistry, definitely having a grip on chemistry. But as far as marine sciences as a whole, and you know, if you're thinking about even something like how do I, you know, work, or how would I find myself on a research vessel like this, there are many paths that get here for sure. We have folks on board with all manner of backgrounds. And within marine science, you of course have marine biology, but we have folks on board that specialize in microbiology, physical oceanography, geology. We've had folks on, you know, looking at the chemical composition of things, mapping, looking at bathymetry and the map of the seafloor. And then for, you know, many of the instruments, if you wanted to get involved with the instruments or machinery involved, of course, engineering, you know, lots of folks get a bit involved with engineering, mechanical engineering, even things like if you're looking at doing the data processing for something like this, getting into data management and data processing, coding. Working on the ship, we have people with backgrounds in, you know, hospitality, from say like working on board yachts. There's more management sort of jobs as well. We have engineers on board, fitters on board who can do some manufacturing and repair on scene. So lots of backgrounds here. But if it is marine biology specifically, the kind of traditional path to that is looking at biology. So that's going to require chemistry, biochem, and then biology, marine biology specialization. But again, people find themselves here from all manner of background. Been going for about 20 minutes or so. People are still kind of trickling in, so if you are just joining us, we are back down 2.5 kilometers down below the surface of the ocean at the East Pacific Rise, near the Tika Vent. And this footage is coming to you live from the remotely operated vehicle Sebastian, being operated from up on the surface from the research vessel Falkor 2. And what we're doing right now is sort of, well, we're doing some suction sampling, more specifically, but just getting some samples, some baseline, and also going to be preparing this site here, the site delineated by this frame, where we'll be placing an experiment that'll stay down here for the rest of the cruise, and then we'll collect it towards the end. And this is the third such experiment. The other two are nearby. Let me actually put up a different camera here. There you can see a box off to our starboard. That is experiment number 2, and experiment number 1 looks much the same. But after we prepare this site here, experiment number 3 we'll start deploying that, and so that's going to involve sealing up the cracks that extend or cross beyond the frame. So there's, for example, a little dent, a little crack, kind of on the side facing us, or both sides facing us really. We're going to seal those up with a like a silicone sealant, sort of do a little caulking job there. And then next we have the mesh box that will go on top, and that box does have its own silicone seal on the bottom. So that'll get pressed down, and we'll try to kind of seal it to the seafloor there. Oh, there's a little crab here. Let's see if the suction sampler gets that or not. Once the mesh box is down, then another box goes on top of that, and that is the staining box. And into that box we can actually pump a dye in that will stain the items that are underneath the box at the time of deployment. I can put this up. This is actually a bag filled with that stain, so we had to bring that down to the seafloor with us. That is about 55 liters of essentially the stain you might use for preparing a microscope slide, for staining something on a microscope slide. And that will get pumped out. You can see, oh, wrong camera there. You can see on the left side, down at the very bottom there of our porch, we have two boxes on the porch. On the left side is the staining box. You can see a little hose kind of strapped to that. And then on the right side is the mesh box. So once the stain is in the boxes, then we'll be here actually for a few hours at that point. And as I mentioned yesterday, we're hoping to possibly be able to give you a little tour of the ship during that time. Just the main deck. We won't be leaving main deck, but be able to show you perhaps the hangar, as well as some of the lab space on board. That is the hope. And then once the dye has set underneath the box, we will be sort of suctioning back up the dye that remains in the box, and we'll lift the outer stain box up, and time permitting, we'll move on to doing some other stuff on this dive. Oh, there's a fantastic career. Yeah. So as I mentioned, lots of backgrounds, and we had someone over on Facebook. I think they are involved in this, but marine seismotectonics. So looking at the interaction of tectonic plates. I actually had the opportunity to work on another research vessel up off of Vancouver briefly as a fellow several years back, and we were there with Ocean Networks Canada, which they were actually installing some instruments to measure the seismic activity in the area as part of an early warning earthquake detection system there. So that's also another very cool one for sure. My own background actually was a degree in parks and tourism and recreation management. So, you know, most of my graduating peers were park rangers, for example, and there are obviously parks along the coast that if you went into that, you're going to be outdoors, you're going to be working. If you wanted to go work at like Monterey Bay Marine Sanctuary, for example, you could be doing that through the government. But there was a course in there about natural resources interpretation, just talking to folks about natural resources, which doesn't occur to you perhaps as a job, but then think about any museum or aquarium or zoo, even a park, right, where you need to present to people about what is there. And so that sort of opened my eye up to that as a career. And I did that for quite a few years and just got hired on here in the past year. And love just talking with people about natural resources. Love speaking with the scientists to understand their work. And then I love sharing that work with the public. So different ways to get here. Someone asking, what's the hypothesis we're looking at with these particular experiments? So this is actually a really cool one for me. I mean, they all are. I think I'd probably say that. frequently. But so the name of this cruise is the Underworld of Hydrothermal Vents, which if you're over on Twitter, there is a hashtag for this, and it is Vent Underworld, and you can check that out. And I'll also post up the link into the chats here for this particular cruise page over at schmidtocean.org briefly. But the hypothesis we're looking at here is that at these venting sites, you do see a lot of the same organisms. You have these, of course, these vent worms, right? Like the riftia worms, these large red-gilled tube worms. You have those kind of appearing along the venting areas, right? So how do they originally get to that area? How do they move from one vent area to the next? Not that the worms themselves move, but as a population, how do they spread? And they could spread up through the water column, but the dynamics of the vents themselves could also act to spread them. So the fluid that's coming up out of these vents, this is heated fluid coming from below the ground, but it actually starts above the ground. It gets pulled in through intake sites. It makes its way down through the earth. Well, not through like the whole planet, but down through the ground, and it gets heated and then flows back up. And where it comes out, where it comes back out of the ground, heated up and filled with minerals and chemicals, that is where you have a new venting site. And so if a larval animal were to get drawn in at an intake near a current venting site, right, an already kind of active site, but it might have a little bit of intake, maybe an older site. If a young larval animal got drawn in through the water there, if it survives the process, if it survives the trip, when it gets expelled back out, it's going to be expelled right at a venting site, right? Because where that expulsion comes out is a venting site. So that would be a great way to transport these animals, or for these animals to be transported to new venting sites. That's kind of difficult to test. So the way this experiment works is we are sealing these mesh boxes down over some venting. We are staining everything inside with a color stain. So everything that's there when the box is put in place is going to have this stain on them, a colored stain. And when we come back after about 10 or 11 days, we are going to look for any organisms inside the boxes or in the area of the experiments underneath these boxes that don't have that stain, right? Which means they weren't in there when the stain was applied. And so the thought there is that we could use that to sort of see if there's any ground there, any evidence there that perhaps fauna is being transported to these venting sites through just the fluids coming up.
It's a bit tight. They were right inside, yes, but the box is a bit thicker than the frame.
Oh, lots of people tossing around like, what about this degree and what about that degree? I'm telling you, there are all sorts of paths to getting involved in marine science for sure. And I mean, honestly, streams like this are fantastic because they really do encourage even citizen science, right? Which just being able to witness these live has really changed the name of the game when it comes to deep sea science in my book. It used to be a ship would leave, folks would kind of wave at bon voyage from the shore, and then you might not see that ship again for months. In like the golden age of sailing and exploration, you might not see it for a year or more, and then it shows up and they kind of tell you these things. They're like, oh, we saw this and we saw that, and it all kind of sounds like tall tales. But now the public doesn't need to wait for the ship to come back. The public can actually join along online, and from the comfort of your home or your classroom or community center, a lecture hall, a camp, dining hall, we can all visit the deep sea and see the science as it's being done and ask the scientists about their work. So let's see, we got some questions here. Did I take dendrology? I didn't really. I kind of took a follow-up to dendrology, forestry science. So, and then someone saying, "Oh, a degree in psych, would that be very helpful?" Or I don't suppose it would be very helpful. I will say that within animal sciences, if you are working with live animals, so for example, if you were doing research that involved working with marine mammals or birds, actually psychology, more specifically animal psychology, but there is a path there, right? It works out. You get folks with backgrounds in psychology and animal psychology because understanding how these animals are thinking and motivations and that sort of thing. Yeah, really, it is an interesting industry. Oh, a question here, and I had actually had this question myself. So someone notes in the comments that a couple papers speak about the worms just being able to release gametes kind of whenever, right? Reproductive cells. And so every once in a while we see a worm kind of like sneeze. And the question is when they do that, there's like some white, like it makes me think of a puffball mushroom almost. And they were asking, are those gametes? I'd asked about this, and most likely not is what we were feeling. We actually think it's probably just some like flakes from the tube itself, because the tube itself is very flaky and kind of sheds. Because the gametes actually need to be released from back behind, sort of, if you want to perceive like the head of the worm. So what we're seeing are the gills, and those are in front of that head, the area that would release the gamete. And the worm would possibly need to come further out past that little stopper, and then they can release their sperm. That sperm will make its way through the water, and if it happens to get to a female, the fertilization on these worms is actually an internal process. And then once the fertilization occurs, it sounded like there was a bit of a pause there in the division of the eggs, but at some point then the female can then release those fertilized eggs out into the water, which was just fascinating to me. I figured they were just broadcast spawners through and through. But yeah, it's a good question.
But so is that like an effect in a big sugar and then
Yeah, think of it as, I guess, dandruff on the worms that we're seeing. As they pull themselves into the tube, right, and their body sort of fills more of the tube, that's going to expel a little bit of water out the front of the tube, and so if there's any little bits of tube flake, I guess, tube dander, that might be what we're seeing, I think, is what the conversation I somehow found myself in yesterday was going towards. All right, so with the experiment, we are now preparing to seal up the cracks in the rock here. So we brought down with us on ROV Sebastian, we brought down these syringes, and these are filled with, again, this is an off-the-shelf product, which I sort of love that. With science on the deep sea, a lot of it's just sort of improvising, or you have to build things that have never been built before, and you have to think about how do you deploy an experiment that's never been deployed before. And sometimes you find things that are perfectly suited for it, but you're just repurposing them for your own use. Sometimes you have to get crafty and make things. So, for example, the boxes themselves are, those are custom made. But the sealant that they're able to use for this experiment is the same sort of sealant you might use to seal a crack at the bottom of a swimming pool. Yeah, someone mentions toilet brushes. That is something that you'll see frequently used. They're good for scrubbing, but also Sebastian has used one in the past to attempt to collect some DNA from midwater squid. So they had a toilet brush. You kind of think of a toilet brush, right? A little bristly, not like the softest brush in the world. And so they were encountering midwater squid and kind of like holding the brush out towards them, because a lot of times the squid will kind of grab onto bits and pieces and check out things on the ROV. And they were trying to get the squid to sort of grab onto the brush and leave some DNA behind.
Okay. First one.
Like a, you know what I mean? No. Basically
Yeah,
covering
I know
this
what it
place
means,
here.
I don't know. It's we can try
Apply it
to
to,
squish out some and then I'll lay it.
yeah.
So we'll do that up in the air.
Basically, we can start a bit behind this white tube.
Okay.
Question in the chat of how many people are operating the ROV. So at this time, we currently have a pilot and a co-pilot. The camera can also be controlled by either of them, but also we give control over to the scientists a lot of times here. And then during launch and recovery of the ROV, more folks come on for that. So it's a smaller team for the direct control of the ROV. Yeah, and then there's usually a technician on here or in mission control to log things. The scientists log their items, the techs kind of log our items. I'll also be doing that occasionally as the multimedia tech, but my main thing here is making sure the streams are running and monitoring the chats.
So let's stop there. I'll see if I can lay that. Good. Okay, if you could once more just point out where exactly you want it. That's where you want this to start. Follow the crack you over. Okay. Just try to push out a little bit more.
So as you can see, the pilots are currently trying to get the sealant down on some areas here. They've found a couple different strategies with it. The first day or so, we were sort of just blopping it down until there was a heavy enough weight to be able to pull the syringe away. And they also found that putting out a little string and kind of draping that or dragging that into position before applying more also seems to work.
Okay, let me see if I can get it
Perfect.
Okay.
Yes, very good. Then we move to this place here. So basically here.
Okay. Okay, try to give that a little squish. A little less hard if you can. There you go. Right there is where we want it. I love that's awesome. Go on to-
All right, here's a good question. So this is one that has come up a couple times. Since we're putting the box over the vents and we're sealing all of the cracks out of the box, won't that like pressurize the box up or pop the box off or float the box off of the site? And the box itself is mesh walled, so the seawater shouldn't build up any sort of pressure, shouldn't be prevented from leaving the box. But the mesh is very fine, so it will capture any fauna or organisms that might come in that water. But the water itself will be able to leave. And then the box is also weighted. It has a little ring of dive weights on the inside to help hold it in place. So yeah, it doesn't appear that the other two boxes have yet been shifted or moved by the venting fluid, but good question.
All right, we're good there.
So the way the controls are being done right now, we of course do have two manipulators on ROV Sebastian. We have a port side or left manipulator, and then a manipulator on the right or starboard. Currently, our pilot is controlling the port side that's holding the syringe, and he kind of gets that into position. And then our co-pilot brings over the starboard manipulator and presses down on the plunger. And the camera is currently a combination of the pilot, the co-pilot, and the scientist. So.
Great. Okay, then we have.
That one.
Yes, you can do that. It's basically almost next to the tube.
That's
It's here's the
this
tube,
one,
yeah.
right? This one is.
That it's
Further up.
nothing. It's, yeah. Yeah.
Yeah.
see here. Sorry.
Yeah, okay. Could you give me a little bit of silicone? Just keep an eye on that box when you reach that far over. It's looking good.
So I've posted up a link in both the chat on YouTube and Facebook. You should be able to find that. It's a pinned comment, but that takes you over to our website at schmidtocean.org, and more specifically it takes you to the cruise page for this cruise, the Underworld of Hydrothermal Vents. But we've been streaming just shy of about an hour or so I'll do a quick little recap here. We're currently looking at live footage from the remotely operated vehicle Sebastian. We're about 2.5 kilometers below the surface of the ocean, at a location on the East Pacific Rise, about 2,000 kilometers to the west of Costa Rica and 1,000 kilometers to the south of Mexico. And currently, what you're seeing the ROV do here is we are preparing a site for the deployment of an experiment.
Yes, so here's one.
That experiment takes the form of a mesh box,
So I think the box
and
should
the
be
idea
over
here is that we are going to place the mesh box down over this hydrothermal, or over this little area of some venting hydrothermal fluid. So if you look at the tube worms there, those white tube worms with the red gills extended out, you can see some shimmering water just above them. Looks kind of like the heat waves you'd see on like a hot asphalt during summer, or over the hood of your car after you've been driving a lot. That's actually where fluid is venting up from below the ground here. And so we're going to place a box over top of that, this mesh box. And then we're going to stain with a color dye everything that's currently here. So it'll all be stained with like a red or magenta stain. And then we're going to leave that box there for like 10 days. And when we come back, we're going to sample everything that's in the box. We're going to sample the fluids that are in the box. We're going to sample the substrate, the rock that's under the box. And we are looking for any organisms there that don't have that stain. The idea here is that those organisms could potentially be arriving at these vent sites in the fluid itself, since that fluid is actually taken underground at other areas and eventually makes its way back above ground at new venting areas. So the experiment here are these mesh boxes to investigate whether or not the venting fluids themselves could be a mode of transport for these vent organisms in their larval stages. If you've never joined us for one of these dives before, we're actually streaming from on board a vessel on the surface, the research vessel Falkort 2. And we are streaming live on both YouTube and Facebook. If you happen to be watching us live on either of those platforms, feel free to type in the chat any question that you might have. And we do try to answer those as we can. For the placement of this experiment, it's a little more intense focus on the experiment. Also, if you happen to be watching from schmidtocean.org, as we do have the YouTube embedded there, and you have a question, just go ahead and click on that embedded video, and that'll take you on over to the YouTube stream, and you can type in the chat there.
Wow.
It appears we have found quite the crack that needs to be plugged here, as the caulking just kind of disappeared down into it.
It's a good scene, though.
Yeah. Yeah.
Move towards this direction, yeah.
Okay.
Oops.
How deep is this thing? Okay.
Or just
Question about the mesh. So if we're taking the time to seal up all this space, but we're putting a permeable mesh box on top, couldn't the organisms get in that way? So the things we're looking for are going to be larger than the scale of mesh we have, but the mesh will allow the water to not push the box up or pressurize the experiment off of the site. But it is a very, very, very fine mesh.
I think I'm better at this subsea than I am in my bathtub at home.
On the boxes themselves, we've on experiments 1 and 2, when we're scoping at them, it already looks like there's been some bacteria collecting on the mesh. In fact, you might be able to make it out just a little bit in this view. So you can see experiment number 2 over there, and there's kind of like a little bit of a white smudge towards the top of the panel, most directly facing us.
then basically
And when we zoomed in on that, that almost looked like some bacterial growth already on there. And that was, I mean, we saw that like yesterday, and that was after the box had been there not yet 24 hours. So. But whether that growth is on the outside or the inside, that's a thing. So inside the box, before we put the box down, we've been draping weighted chains with some sponges on them, like bath sponges, and that actually provides a porous substrate for organisms that look for that sort of substrate to settle out on. And there's also been another substrate on there, sort of like a plastic or acrylic substrate that has successfully been used before to recruit tube worm larvae out of the water, and so that's in there as well. And so we're leaving that underneath, then we're putting the box on top. When we come back, the first thing we're going to do before we even lift the box off, because what if there's stuff that came up in the vent fluid but it's still suspended in the water, we're actually going to kind of pierce through the side, through the mesh, and sample the water inside the box itself. Then once we've sampled that, we'll remove the box, we'll collect the sponges, we'll collect the tube worm recruitment. There's also a temperature data logger in there that'll kind of record the temperature throughout the experiment. And then we will try to sample the substrate itself, the rock that the box is actually down on. So lots of different sampling going on with this experiment, just to try and cover all bases.
Nice.
Great.
Well done. So eventually we put enough of the sealant down that it finally started filling that crack.
This one, this one, this
That was an
one.
impressive one.
That bottom one.
Bottom is so this is that from here, so it should be here. Need to zoom in a bit. Because I'm not so sure what is marked here, if this is really needed. Do you want to use the camera or should I?
Sure, go ahead. Right there, okay. Give it
I'm
a
not.
little-
Wait a second. That's not. I think that should be fine. Like here's the mark, I think. Oh.
Yeah. But the frame is just a little bit up from
Yeah,
there.
from
So right there, exactly. Yeah, that's right where
yeah,
I'll land.
yeah.
Okay.
Just a little bit. It's not needed. Not enough.
Okay. Good. I'm just correcting a little. It's gonna pitch over whenever you push down on it.
And watch out here in the back. Ah no, sorry.
Yeah.
Forget about it.
Yeah. Okay, stop there.
Brought down a couple of these syringes.
A bit, so
It
it's,
seems like we've typically only needed
must I
one,
think
but
of it a bit
always good to
upwards,
have
a
a
little
bit more than you
not
need
down.
in case every crack you're
Sorry,
trying to
but it's
fill
like
is
here.
super deep.
Okay, I thought that was
So
exactly
like
where
here, yeah. Just
you wanted to go that way.
here, this is good, but like if you, it's good. It's like from here to there, this area here.
Yeah, it's about to break.
Mm.
Let me, yeah, we're gonna have to.
in the foreground here at the bottom right of the frame, you can actually see the bottom, one of the bottom corners of the mesh box, and you can see there the clear silicone seal that's on the box itself. And as we saw from one of the earlier experiments deployed, that's very squishy, very soft seal. So hopefully it conforms more to the sea floor here. The sealant that we're currently applying is more for the major features, the cracks and the larger depressions.
It's also kind of handy because it gives me an elevation reference.
So I believe after the sealant is applied here, then we'll move on to placing the sponges. If you've been around for some of the earlier dives, you've seen these colorful bath sponges. Kind of another example of something off the shelf that actually works great for this experiment. But obviously it's not really its intended purpose when you see it on the shelf at the store there. It's one of the aspects I really enjoy with marine science, deep sea science in particular. It seems like it's kind of the repurposing of materials from what you would anticipate as their use.
Okay, good.
Good.
So the frame.
I just assumed that they would get.
And again, after placing experiment number 3 and staining it, time allowing, then we will be revisiting the sites of the student experiments that were deployed yesterday. But once all 3 of these, well, we'll have to go get that. But once all 3 of these have been deployed, these 3 experiments, then we will still continue to have dives, but those dives will be a little more mobile, I believe. There is a example of one of the issues that we've kind of encountered with the syringe is just we've been using the plunger itself to sort of fit down in those coral quivers. Or those over there, I guess, aren't coral quivers. Those are push core quivers. But once we've used the syringe and the plunger has moved significantly more in, there's a lot less purchase there for us. So we'll have to grab that afterwards, but no worries. Just loosening the bungee cord holding the mesh box down.
But the sponges in the bigger tank.
But before we put that box down, we're going to deploy the sponge chain. So these are the aforementioned coral quivers.
RV bridge.
Go ahead, Bruce.
It's just a bit of weather, so I'm just moving us around maybe 20, 30 degrees to port.
Okay, copy that. I'm gonna give you a small vessel move for that.
Thank you.
All right, so if you didn't hear the call there, we're having a little vessel move, and there is a bit of weather over us right now. From what I can see, it looks like our stream is still holding on, but here's kind of what it's looking like topside right now. So if we are buffering at all for you, apologies. Hopefully we'll be through this soon enough, and we can get back with a stronger signal, but we'll see how it goes. Yeah, weather's not too bad, but just the rain by itself sometimes will give us issues. So, fingers crossed. This always makes me think of Sebastian doing like the handkerchief chain trick. Alright, so you can see the sponges, and then at the bottom is that acrylic tube worm recruitment instrument, as I was mentioning. And so what that is, you got these the acrylic, and there's little holes drilled in the plates as well. And this actually has a history of being used to recruit tube worm larvae. They like those little tubes, those little holes. They'll kind of cruise on down in there. And then when that comes back up, if there are things that have been recruited, to look at them under a microscope or something, you can just take that little nylon bolt out of the middle, and everything is nicely in a little sheet for you. We have the sponges, and then there's also a temperature data logger there as well that will record the temperature during the deployment of this experiment here.
You need that off. Is that what you're trying to say?
That's good. It didn't seem to go there. Without saving it.
Okay.
Please, a great job.
We can hide it from that.
Yes. Sorry, that was logging you.
Yeah, and we've got a couple different networks typically that we can bounce around with, but the streaming we try to keep on a network instead of moving it around constantly. So if we do start to get a little spotty, I will fiddle with settings here, but sometimes there's only so much we can do. But if we do get a bit spotty with the internet, once that gets back to full strength, we will hopefully be bringing this back to you. You can see there a little squat lobster that's hitched a bit of a ride. Not uncommon for us to see in these areas with a lot of squat lobsters, as they're pretty keen to propel themselves up into the water column, and then they sort of parachute back down. They spread their legs out and just like glide like a skydiver, and frequently that sees them landing on parts of the ROV. All right, the pilots are consulting a still frame of where the actual frame was, a little video still frame of where the metal frame was, while they work out the placement of the mesh box. You can see the, again, the silicone seal on the box itself. It's currently turned so that we can kind of see up through what will become the bottom of the box. You can also see the ring of weights in there to hold it down. Yeah. All right, a little bit of damage to the seal there, so we'll have to see how it presses down. It is pretty soft. Currently just taking some frames of the seal here, so we document its condition. Pilot speaking with scientists. Sounds like we're going to put it down and see what it looks like. All right, so scientists are currently looking at where the frame was and how that particular corner will be laid out.
I think it's not. I think it actually pushed down. Didn't break off.
Seems to be discussion that
I'm wondering what is better. So I think it could work out. Just
it may work out because they're going to kind of
also
compress the seal down. And again, it is pretty squishy.
I think it works actually because it should push outwards. If we
But
want to blow before, then we need
to do it now.
recover this again, and then we might damage it.
No,
more.
like we either like do it now or not. It's
Yeah.
like
Yeah, because you cannot. They needed the two arms, so
Yeah, yeah.
I think the danger is put it back somewhere now and to damage it more. So I think
no,
I
like
would
either
just
you
put it on.
like the box you mean now.
Yeah.
Put it on.
Yeah, yeah. It's
Well, this box we can put upside down on a rock, right, where we won't damage it into the silicone.
I think
It's not
I
because
think
it
it's
will.
not bad.
I think it will be pushed inwards, and it's so sticky and so soft that it will, seal itself.
yeah.
When it's not breaking off. I think when it's breaking off,
Sure.
I think it's gonna be. This area look is yeah, however this is not really clear, but it will just squeeze together. Come on.
think it has a high chance of squeezing back together after touching it. It's so
Yeah,
soft.
yeah.
And it kind of wants to go back to its
Right.
original
Yes,
form.
yes,
So
yeah.
when it gets some support by the rock, I think it will.
We'll
But
find. Yeah. Okay.
we'll just have to look afterwards.
Good. So that's
All right, so it sounds like we are go for deployment on this one still. We'll just kind of scope it afterwards and see what it looks like.
These are okay as far as that's the onboard kind of flatter. Unless I need to, I can't go back far enough. Yeah. Which, looking at the orientation, I think you're right. Because I can't get it out of
Work around it. I think that might be a good spot for you to see what it's like. And that's like you need to rotate it a little bit, counterclockwise. Right about there. See here, you have one mark there, and the other one is here.
I see the marks. I see.
Yeah, that one and this one.
They were quite outside. So here, then like here, and
Yeah, that's a corner. That's a corner right there. So, yeah, that corner here.
So the pilots actually marked the ground a little bit with our chisel. So currently they're trying to line up with where the marks were.
You're way too close. It's because of the angle of this camera. If you look at this, so you're setting this down, you're over this muscle. You're about to land all the way down here.
I'm just watching the shadow of the marks. Okay, you know what I mean? Like,
this is too.
whenever it's more clockwise. I'm asking you if you think more clockwise.
Yeah, it's about the same as it's been. All right, so that's the mesh box down. And now we'll be kind of scoping around with it.
It's a little low, but
So the marks,
the marks were on the outside of the frame.
yeah, and they still are, I think, right there.
You're like on it.
Okay, and the left one is a little bit closer. Yeah.
Check out
Flare up.
from the other side.
Yes, here. That damage corner is here since
Cool. Pretty good on that corner. Yeah, got it right on that corner. Sweet. All right. Yeah, they're pretty happy with that. So that is the third experimental mesh box down, but now we're going to be moving on to the dyeing portion of it, which means we have to deploy another box on top of the mesh box. And once the second box is on, once the staining box is on, then we pump a staining dye into the whole system under that second box, and then we'll be sitting there for a while.
Yeah. What? Got some damage on this. Yeah, I saw that. Really? It's just gonna make it much harder to get that stain on.
Is that how far is this thing?
Yeah, about enough. Close.
Yeah, I'll have a look at channel top.
Yeah, mesh is intact. It's just all right. Got a bit of a handoff here. Jill and ROV pilot Alex are swapping out, and we've got Chris and Russ swapping in.
Warm sea.
We're gonna look at this side with the handle on it for us.
So it looks like there's an area with a little bit of acrylic sticking out. The mesh is intact, so that's not a problem. But the staining box just needs to slide down over top of it. So we'll just see what we see. Yeah, as you can see, this is a good demonstration. After the box has been here for a little bit, that silicone seal is squishing. It definitely gets some compression to it. So that little bit of damage that we had on the other corner is likely just getting mushed together at this point. So. Scientists are pretty happy with that. So onwards to the staining. All right. Here's a fun question, and honestly, I just learned this, so a fun answer. We had a question of when the tube worm larvae are released, are they neutrally buoyant, or do they sink? So we talked earlier that the sperm is released from the male tube worms here, and that's a broadcast. But then that makes it to the females, and the fertilization occurs internally. There's a halt in the division there after fertilization occurs, but then the female can release out the fertilized zygotes, and once they'll start developing again. But when they're first released, they actually have quite a lot of yolk at that point, and so they are actually positively buoyant to a degree there. But the more they develop, the more they grow, and the more they use up or ingest their yolk, the less positively buoyant they'll be. And they'll actually become negatively buoyant at that point. And so then kind of settle out. So if they're up in the water column and they happen to settle down where there is no venting, that's it's bad news for them. So that's why one of the things we're looking at is, could the water intakes, if one went down into an intake, if they got pulled by a current down into that intake and then kind of got shot up out of a vent, is that a way that they're making it to these vent sites? But that was really cool to learn that because of their yolk, they're beginning positively buoyant, and then as they grow and as they use up their yolk, they lose that buoyancy.
I had an idea. We can stick it in the back out, just the portion on there, like that.
And so here we have the staining box that's being prepared.
Slide it in there.
So this box is going to nest over top of the mesh box, and the dye will then be pumped into this box, the stain box.
Don't be in the way. Put it back on. Yeah. Make more, we figured it out. I would not, I don't know.
Well, this one's got a cable.
Yeah, I just don't know if picking it up with that, it'd have it swinging on the porch.
Well, I need to get it out. Yeah.
Okay.
And then we can lay it down.
Yeah. Yeah, that's what I was saying, I didn't know picking up by the rope.
Oh, no, this one.
Yeah, that's what I was getting at. Got a good view on the sit, just creeping in there.
Should be too bad.
Yeah, she probably.
This is good.
Shoulder straight up, there you go. Alright, now I'll let you get it to where you're comfortable holding it, and then I'll move this in. You know what I mean? Whether you wanna.
Well, I was gonna probably put it down to the ground.
Yep.
And then re-grab it.
Yep.
So it's flat.
Yeah.
Lay it down nicely.
Whatever you're comfortable with.
So when the zygote becomes divides the right side, it becomes a larvae. The larvae settles, and then the larvae starts to see the symbiote that has the other layers. where the symbiosis
Yeah,
is staying.
that's perfect.
All right, so continuing on with our development of the tube worm, right? So you have that fertilized zygote released by the mother. Zygote's positively buoyant. It has this yolk, and it's dividing, it's using up its yolk, and it becomes larvae. At some point, it'll settle out. It'll be heavy enough, it'll sink to the bottom and secure its spot. But once it's a larvae, that is when the symbiont, the bacteria with which these animals have a symbiotic relationship, that's when the infection will occur there. So that's another thing that could be getting moved through these vent flows is the actual symbiont bacteria. You know, if you are a tube worm and you happen to be a little larval tube worm that just settled down next to a venting site and you're really needing to get infected with that symbiont before your digestive tract atrophies away and you have no way of feeding yourself, it'd be great if that bacteria that you need just happens to get delivered to the site via the venting. So lots of interesting possibilities here, and hopefully these experiments will give us a bit more of an understanding of the potential role that venting fluid plays.
Where's you want to be? How's that?
Yeah, that's really good. Just maybe tighten a little more.
Right.
Just drop it in. That's good. Steel.
Nope. Nope. Yeah. Good. I think we're going to sit here for a while, so that's a good amount. Yeah, I just didn't want to go too hard because we're going to be sitting here for hours. Don't want to get hot.
All I want to do is take over this.
There was a Whitney Houston song playing. There was a Whitney Houston song playing upstairs before you came down here. It's a great song.
I'll adjust the pitch when I get there. I just need to change my wrist. I need to get up some of that. It's all this.
Yeah, bacteria living down in the substrate itself is certainly a thing around here. And as they've been bringing up little rock samples, part of the procedure there is kind of crushing some of that rock up so that you can use different methods to try and locate an ID. Our little weather report here. It's still raining, still overcast, but the internet seems to be holding strong for now.
square you can. Pretty south as well.
Let me just get this out of your way so you can see your arm in HD1. Might catch in the back corner.
And I'll just adjust it.
There you go. Just yeah. That's the angle. It just needs to. Now you just come down a little bit. Might come back here because it's just spinning up. Might be alright. Yeah, hold on. You come, there you go. Might be alright. Go slightly to your right. Ever so gently. Alright. Alright. Yep. You good. You go, you're on it.
All right, so just nesting the staining box down. There was a little bit of damage on the outer acrylic, but that's fine. The mesh was still intact. We were just not sure if that outer damage would impact the fit here, but it seems like it'll be okay. There is a little bit of play on that particular fit, so.
Nice one.
It's easier than you thought it because otherwise probably it's getting a bit tight. So, I tried.
Alright.
All right. Well done.
Okay.
Well done.
Now the pressure.
Yeah. So now,
Almost did.
now that that's in place, we'll be turning on the stain, which is it's actually stored in a bag on the aft port side of the ROV.
Yeah, we'll do the same. So, tell you what, Chris, I will give you the port
Yep.
because it's easier for
That'll
you.
make more sense.
Not with Underworld, eh?
It's not like totally straight. It's a bit more. Do you see that? See a bit of this
Mm-hmm.
a little touch from the right side
Yeah,
Very
yeah.
gentle, very focused sides.
Probably it's a good idea to get the other arm on there too, though, just so it doesn't move too much. You know what I mean? It just lays the arm on the top. That way, if I touch it, it doesn't disappear. Do I need to?
All right, just gonna get the box to settle all the way on, but pretty good so far.
You can just get the rope and kind of adjust it a little bit.
Yep.
Just
Sorry,
give it 2 seconds. Let's try
didn't
it.
mean to leave you on the stain bag there.
You have to look down at it and see.
Yeah, see, you can see it's not level. Just you hold on there, Chris, and just see if this will move it. Here you go.
Alright. Push it down. You want to look first?
Yeah, have some.
Yeah, that's better.
Push it once more down.
Yeah.
Question about the effects of the pressure on the bag. Bag's just filled with a fluid. I mean, there might be like an air bubble in there, but it's a flexible bag. So if there's a little air bubble in there, that'll just get compressed out. But the dye itself isn't a problem. It's not gaseous or anything. So being a fluid, it's not as dramatically an issue as far as the pressure goes.
Alright. On. So ready.
Ready to expand. It's brilliant. Give me a little light.
Come on, just a second.
Probably.
I watched the camera.
Okay. Do you want to push your arm forward so it don't scratch it? Just in case. Oh, yeah.
You see that it goes down or out, goes up.
All right, so that's the dye spreading in there, and again, that bag has something like 55 liters of dye. So we're gonna sit here for a while and just pump it in there, so that it really just saturates the entire inside, and then we'll actually suck water back up, just kind of sucking up a lot of the dilute dye there. And then we will take the dye box or the staining box back off. And what will remain is the final placement of the experiment, and that'll be left there for over a week, I think is the plan right now. And then we'll be back.
This is super fast today. Not that they've been like the atoms, not that it was. Yeah. Every day we are quicker. Or you are quicker, not we. You.
People are inspired.
So, what are we doing tomorrow then, since we're done with the 3 boxes?
Tomorrow we go further north.
Okay.
Explore the area. We want to go up to Lovely. So you have the maps. So right now we're hanging out here, and we have been once to Teddy or twice. And tomorrow we'll also start here, then go there, and then wait.
Sure.
Ah, new map.
That's some of the names like Perseverance, Teddy Bear, and other ones are like
Yeah.
Eastball.
Eastball. Yeah, but it's a wall on the east side.
We go here for the school, we go to Crabs Bar. No, this is the day after. Here. And our side is here, up here. This is not in here, but this is the passage. 7, 8 is our side.
So tomorrow we will collect rocks in this area, but it doesn't take as much time as all the tube rooms. And then we go
Oh.
north.
We're taking our frame home with us today.
Sorry?
Bringing the frame back with us today.
I think I don't know, at one point we have to bring it back.
Say again.
No, the frame, we don't need it tomorrow, so we don't need it anymore now.
Yeah.
But we have to bring it back at one point, whenever it's easier, probably for the setup of the porch.
Actually, I think the only time we need it.
Huh? That's something else, not me.
The only time something I think we need it is when we come get it and later open the house. To lay it on or something.
Then we leave it here.
We could just
We
leave
leave
it
it.
here. I could leave it here.
Mhm.
We
Yeah.
don't have to. It's the only
Yeah,
other time
yeah.
because the basalt we don't need it
No.
here. That's the only time. Yeah.
Before we lost the syringe, or like not lost, like it when they tried to put it into the back in the push cord, it tangled over on the left side. So probably if all the staining and everything is done, we can have a look where it is.
Can't trust them with anything. Can I look around right now, or you gonna wait till it's? Can I look around right now, or is it stay focused on this film?
I think it's fine to look around a bit. Yeah, we already know that. Yeah, it's good.
Looks like a air conditioning unit.
We're just standing for half an hour today.
35 minutes, and then we stop, and then we wait for 2.5 hours, and then we come back.
Ah, okay.
So total it's in a 3.5 hours, or 3 hours 40 minutes. So it's good. Oh, a small tube post.
Give me this one.
So tiny.
This is
This
They
is
get
super
super
large.
these
Yeah.
days. Yeah. Yeah, so they. You showed me one that was like this. No?
Smaller, like a millimeter.
I can calculate the size easy. And I just have to put a scale on it. Yeah. But it's so basically the other scale. Which is good. This size the larger ones.
Pete Gerges is watching.
Yeah.
Durgus? Hello, sir. You rented us a taco truck, so you'll always have a place in my heart.
Sorry?
You rented us a taco truck in
Yeah.
San Diego, so you'll always have a place in my heart. It's adorable. It's so little. Let's see what's going on out here.
Hello, everyone. Good afternoon, good evening, morning, and good night to anyone, no matter where you might be tuning in from, watching the stream here today. My name is John Fulmer. I'm one of the technicians aboard here. Just going to take over for Alex for a little while. You are viewing live footage from ROV Sebastian at 2,500 meters beneath the sea surface in the East Pacific Rise in the Eastern Pacific, as the name implies. We are streaming from our research vessel Falcor 2 of the Schmidt Ocean Institute. If you've been following us along for a couple of for this expedition, as you've seen on the screen and as Alex has been relaying, but if you're just joining us, this is a dye box. So there's a mesh box that is affixed to the substrate of the sea floor, and then another one over top. And this is just red dye, non-toxic, biodegradable. So don't be alarmed by the color. It is looks like there's some horrible things going on in there, but it's just in order to dye any organisms that are within this this closed-off space, and then the mesh box allows seawater to flow freely through it. So once everything is dyed in there, we will be having to sit here for about 3 hours to let the dye soak into all the organisms there. And then once that is done, we will remove the remaining dye. We'll remove the outer casing of the box, and then it will look much like the one on the right side of the screen there as the dye starts settling. And then when newer organisms come up through the substrate, they will not be dyed, and we'll be able to determine how they have entered this area, because they can't get through any other way than up through the ground. So it's the purpose of this experiment. Being that this does take quite a while for the dye to set in, and we can't go anywhere because we have to stay attached to it with the pump, we will be doing some tours of the ship later. Some of the scientists have graciously offered to walk people through the experiments that they're doing and the findings that they may have discovered so far, what they're hoping to see. You'll be able to see a little bit more of the ship and get to know some of the amazing science party that we have on board for this expedition. And everyone, just a heads up, we're going to be going quiet here for a little while to do some testing, just to make sure that we do have comms for the other sections of the bridge, or other sections of the ship.
just saw right there, that is the. Yeah. It's, you know, getting itself out there like that.
Yeah. Is there a spur?
Right, everyone, welcome back.
Could be.
So you can see some of the rifty off in the distance here, and the dye box in the inset.
It's a cool shot.
Frame grab of that one.
Yeah.
Okay, come back up.
Boom. And if anyone was wondering what that was, there, the shackle that we saw, that's a marker from a previous expedition, being that this is quite a popular and unique area in the Pacific. It's had quite a bit of interest. And many of the researchers we have on board here have been here upwards of 5 times already in their career. Okay, yeah, that sounds good. Just let me know when it's ready to go. All right, everyone. So in a moment, we'll just be cutting to a roaming camera out on the aft deck. It's a little bit wet right now, so it's going to be like a a field reporter in a snowstorm or something. But Alex Havens, who you guys have been serenaded by for the last few days, who's doing an excellent job on the stream. He'll be on the aft deck and walk you through the ship RV Falcor 2. Sorry, Alex. Okay, you good now? Audio is live and come to the shot. You're welcome.
One. Are we good now with our audio? All right, always something. So hello and welcome to the aft deck of Falcor 2. And so if you've been trying to put a face with the voice, my name is Alex. I am the multimedia technician on board for this expedition. And as you can see, the rain isn't too bad out here right now. But we are indeed at sea, again, about 2,000 miles to the west of Costa Rica, 2,000 kilometers to the west of Costa Rica, and 1,000 kilometers south of Mexico. Getting rid of my American non-metric ways here, slowly but surely. But we're gonna kind of do a little tour of main deck, and then we'll toss it back and see where the stream is. And at some point, we're hoping to also go through some of the science with the scientists in our main lab. So this is the aft deck. It is pretty large, but it's raining out here. We don't spend too much time, so we're actually going to head into the hangar. Now, because we are streaming, because the RV's in the water, hangar's going to look a little empty. And it's a little noisier here as well. So in this main hangar here, we actually have two large doors. So to give you an idea, the door over here is closed, but the door over on this side is open. And this is actually where we sent ROV Sebastian over the side earlier this morning, down 2.5 kilometers to the bottom. So you can see there actually the umbilical tether that is connecting all the way down to Sebastian. That comes up into the A-frame, up and around, and then down under us to a winch. And that winch actually has a total of just shy of 6 kilometers of umbilical on it. So that whole signal has to travel down the tether, any commands that we're sending ROV Sebastian, and then the footage and any data Sebastian's collecting for us gets sent back up. And that seems like it should take a long time, but it's all on fiber optic. So basically speed of light, right? It's very quick. There's not really any perceptible lag when the pilots are controlling Sebastian. But when the dive is done and we bring Sebastian back up to the surface, we'll land him right here. And then all the scientists are kind of just waiting. We'll go over to their side here in a moment. And they just wait for the ROV to be down. They come in and grab samples, and then we'll take a look at the labs in just a little bit. We have some workbenches here as well, because in between dives, there's always some maintenance to be done, right? Every time we're diving, you might notice that the pilots talk about like, oh, we should move this here, we should have this grip here instead. So it's constantly a little bit of an experiment of its own, where they're working to make Sebastian's operation much more efficient or more smooth, or just figure out how we can get more out of the ROV. But as I mentioned, there's a door on the other side. And so there's some work being done over here right now. We're not going to go too far, but this is where the CTD is. So this is our oceanographic device that we can drop over the side. It also will go out on its own little J-frame, not an A-frame, right? An A-frame goes up over the top and pivots out, and the J-frame just goes about halfway and pivots out. So this will go out on a J-frame, and this is measuring values in the water for the conductivity, the temperature, the depth of that water. And the conductivity is a measure of the salinity, really, of how much is dissolved in that water. So this is a really important tool for us. Now, you'll notice that when you're watching the stream, like right now, down in the corner, there's actually some numbers there, and those numbers are actually from ROV Sebastian's on board CTD. So we have that instrument on board as well. So the other thing that we have here on the CTD we have is a rosette of Niskin bottles. Now you've heard us talk about Niskins on, again, Sebastian. Sebastian has like a handful of Niskins, but as you can see we have quite a lot of large Niskins on the CTD here, and when we deploy those down we can actually collect up to 24 samples of water there. 12 liters each on those 24 Niskin bottles. So we're going to head on in. After the samples come off of ROV Sebastian, then it comes into the lab space. So we're not going to be pestering the scientists just yet, but we are kind of swinging through. This is the first little area. This is the hydro lab. Out there next to the CTD, there is a dirty wet lab. That's like if you need to wash off push cores or get the sediment out of them or anything like that. But this is the first of the more established labs that you might think of. This is the hydro lab. So they're just doing some work over here. The cold lab, we might peek into that. So this is the cold lab. We have lots of fridges, lots of freezers. Some of the freezers go down to -40, but if you are needing to process a sample and you're needing that sample to stay cold, then that is where the cold lab actually comes in. So this is essentially, I'm just going to pop in there real quick. It's just a walk-in fridge. We can bring the camera on in here. Close that door. Excellent. A little dark in here, but that's all right. So this is the cold lab, and right now it's down about 5C, 5.6 it looks like. It'll get down to about 4C ultimately, and we've got lots of shelves in here for storing samples, but then there's actually countertop as well where they can take those samples out and process them right here, all while they stay nice and cool. So this is the cold lab. So far we've got our dirty wet lab, our hydro lab, and the cold lab. So let's go see what else we've got. There we are. All right. So after the hydro lab, then we kind of come to this little vestibule here. We've got the wet lab and the main lab. So let's check out the wet lab real quick. This is one of our smaller labs, kind of about the same size as those previous two labs, but with a little bit more instrumentation for actually doing work with the samples. For example, we have extraction hoods over there. So if you're working with something that you kind of want the air getting sucked away from, you can do it in there, in those extraction hoods. But we also have lots of counter space back here for doing all sorts of work with the samples themselves. So this is the wet lab. Hydro lab, wet lab. Dirty wet lab was all the way out by the CTD. It gets a little confusing sometimes. All right, next we've got the main lab. Now, because the ROV is in the water, some scientists are doing some stuff right now, but a lot of scientists are also resting, because once the ROV comes up, that's going to be a busy time, right? We're going to get those samples off the ROV, they're going to start processing those samples. So this is the main lab. As you can see, lots of stations here. A lot of this gear is set up by the scientists themselves. They actually bring this gear with them. We can provide things like the large freezers that aren't going to be great for transporting. We have some more fume hoods in here. We have an ice machine if they need ice. But a lot of the more specialized gear, stuff the scientists know they're going to need that, you know, not everyone needs, for example. It's very specialized to their work. That sort of gear they can actually bring themselves, and we'll load that on the ship and help them get it all set up here in main lab. So we'll kind of work our way down the lab a bit. So we'll be back in here a little bit later, and we'll see if any of the scientists will walk us through their work. You might hear a slight echo, and that's because we actually have a screen. I'm going to have Tyler look at it, and you're going to get the weird tunnel vision. All right. So we do have screens around the ship. If there's anyone who's doing work and they need to know what's happening on the ROV, or maybe they are keeping an eye out for a particular thing that we need to sample or collect, that's an opportunity, right? We can just put that up on the screen anywhere, even up in the mess as we're eating. We'll have the ROV dive up on the screens, and people will be able to see what's going on. All right. So now we're in a very long hallway. If Tyler looks back down that way, that actually goes all the way back down to that sort of vestibule area we were in when we first went into the wet lab, and then the main lab from there. Down this way we have one more of our labs on this side, so we'll peek in there.
Hi everyone, and as Alex takes you into the seawater lab there, he's doing a great job of the tour. Just to keep everyone in the loop here, we are now currently turning off the pump. So we've filled all of the dye in, and you can see one of our ROV pilots, Russ, is using the manipulator to turn the pump back off. And there we go. So the pump is off. Alex is standing by waiting, but it'll just be a couple more moments here. And you can see there that the bag that was full of the red dye is completely depleted and emptied. All right. And that is off. And now we'll get back to the slightly more exciting stuff of the tour with Alex Havens.
All right, so as I mentioned, this is another of our labs. This is the seawater lab, and by far I think this is like the most CSI, if you're watching a show and they want to show you a lab, that it looks like this, maybe. A lot of times scientists go, they don't look like that. This is all sorts of weird pipes and running flow meters and or flow indicators, the tubing. If you need to do something with the seawater itself, we can collect seawater in. We have the ability to like cool seawater. If you're monitoring microplastics, we can put you on a loop for that. So Seawater Lab, as the name implies, is for looking at things directly with that seawater from where the ship is traveling through. The Niskin bottles that we saw on the CTD or that Sebastian might have, those bring up seawater as well, but that will typically get worked on in a lab down that way. This is for actually drawing seawater up actively on the ship for the most part. We also have a liquid nitrogen generator in here. If the scientists really need to cool something down, we've got it right here, so we can provide that on board the ship for the scientists. And then there is a lot that goes on. I'm just going to open this real quickly. This is one of our least populated racks. So just to give you an idea, every room on the ship pretty much that's being used for science has a rack of some sort that has machinery or computers that are helping the scientists do their work, helping us push the signal around the ship, helping us push the signal to you, helping us store terabytes of data every single dive. So there is a lot of computer work. We're actually going to see that in our next lab, which is going to be the computer electronics lab. So let's go take a look over there. All right, so we're back down this way now. So we're headed towards the back of the ship, which means over to your left, that's actually the starboard of the ship, and over to your right now, that's the port. It gets confusing sometimes, but folks catch on pretty quick. So over this way, we are going to head over to the computer electronics lab. Now, on this particular dive, this area is pretty well mapped, this particular cruise. This area is pretty well mapped, which means we don't need to be doing a lot of mapping, but that is something that we could do. Or the computer electronics lab can also be used to run our equipment, where we're hosting this stream from, but mapping, looking at the CTD, operating the CTD winch. The scientists can set up in here as well with all of their computer gear. We actually have our multimedia coordinators are set up back there. They're the ones that edit those nice clips that you see on YouTube when we have weekly updates or when we have 4K highlights from the ROV after a cruise. That's all their work as well. All right, there's one last lab that we're going to swing into, and it's going to look kind of different, but at the same time, it's probably the lab that you are most familiar with in a way for this stream. Oh, I guess before we go in there is one last little lab. That's our robotics lab. It's just a smaller lab. We don't need to go all the way in there. This actually leads back through to the hangar. So that's a little sneaky lab that's kind of tucked out of the way that I forgot about. But this goes directly out to where Sebastian is. And so as I mentioned, the pilots a lot of times will make modifications to Sebastian. And when they're doing that, if they need to solder something, for example, they can go do that in robotics lab. That was the sneaky one, almost got away from me. So we're going to head in here now. And this is the mission control room. So this is actually where we are controlling Sebastian from, and this is where mostly we're streaming from. We've got John putting another face to a voice over in the corner there.
Hello, everyone. And I can pull this mic up as well. Hopefully we're not getting too much crosstalk. Yeah, welcome to Mission Control, everyone. Great job, Alex.
So let's just kind of take a look around. Obviously, there's lots of screens, and many of the screens in here are showing the same thing. But again, if we're doing something else, like if a CTD was running, then our marine techs could actually be monitoring how that might be going from here, while another marine tech might actually be dropping that CTD down and bringing it back up. If we need to bring up our bathymetric data, if we have a scientist who's using a particular instrument or needs to be looking at a particular data set, that'll happen in here, or logging will happen in here as well. So from the front, we have our science chair over there. So currently we've got Sabine in there. And so whenever you hear a scientist narrating, that's the seat. And then the next ones, we've got our pilot and co-pilot seats. And so for today, we do have a co-pilot in here, but right now they're out because we're not currently piloting. We are sitting on the bottom, so they're probably grabbing lunch. I would imagine we'll have someone pop back in while we're in here, actually. Oh yeah, so Tyler is going to show you up close the manip controller there. So you might remember on last cruise, that was Tyler's first cruise out here as an ROV pilot, actually, and he was learning how to use this controller. So John's going to demonstrate it there. So it's a very odd looking controller, but basically what you have is a miniature version of the manipulator. And so as you are moving that, it will move the arm in relation to how you're moving the joints of it. All right, we're going to see if John can be trusted with the manipulator. All right, so there we put the arm out a little bit. You can see it there in that picture-in-picture we've got going on the stream. And so that was just kind of a quick little tour of the main deck, from the aft up through the labs. Up above us are more the living areas of the ship. We have cabins, we have the mess, the galley, a lot of personal space up there, right? So this is the area where a lot of the work happens. As we said, you know, when Sebastian comes up, it's going to get a lot busier because Sebastian comes up with samples. First of all, the pilot's got to get out there and recover Sebastian, actually bring it back on board. Then the scientists go out and get their samples. Then the pilots are working on anything that they need to fix or modify on Sebastian. Pilots are busy processing those samples for quite some time. So that was a quick little tour, and I think we'll probably toss it back over to the stream in just a moment. John's kind of caught now in a conversation, but we're going to toss it back on over to the mainstream. And I hope you enjoyed this quick tour. We're going to try and give you a little more insight to the experiments being done and the work being done on the ship in just a little bit. So stay tight for that. And until then, we'll take you back to the stream.
Great work there, Alex. Thank you very much for the tour. I'm sure everyone out there in Divestream world really appreciated that. And later, as Alex mentioned, later on we'll be going through the labs with the science party, and they'll be able to show us some of the experiments and get into the nitty-gritty details of everything that they're working on here. But that's a nice little overview to kick things off. And with that, we will go back to the box.
Wie lange ist die geworden?
14 Uhr.
Mit
Stunden und dann kann man so zupfen. Immer 2,5 Stunden gewartet, dann haben wir das eine Stunde zu suchen.
Also wir können tatsächlich beide gehen.
Genau, ja.
Ja, wo ist die Person, die dort sitzen soll? Dann Leser. Aber das war alles ja.
Ja, das einer soll wahrscheinlich da sitzen von uns von
Ja, aber
das ist.
von uns da ist das selber oben gleichzeitig essen. Selber und im Frühjahr das habe ich auch schon. Und da brauchen Sie sich mehr Zeit, dass wir hinschauen, wenn es heute noch drauf ist, damit man was wichtig ist. Ne?
Ja, also heute ist noch drauf zu den Kindern-Experimenten schauen und
Ja.
noch Samples nehmen. Vielleicht können wir
Samples nehmen.
also eher da wieder einfach wieder ein Suction-Sample von Muscheln.
Okay, das dauert
dauert
nicht
10 Minuten.
Minuten.
Und das war's eigentlich.
Okay,
Also Rockcracker probieren, aber das braucht man nicht heute machen. Das ist dann, den lassen wir unten liegen.
das muss man jetzt nehmen.
Genau.
Das machen wir. Also wir haben eigentlich kein große Bar drauf jetzt schon.
Ja,
Ne?
bitte?
Dann haben wir wieder uns.
Den Bar ist drauf, ja sicher. Den haben schon die Leute. Das ist ein Riesending.
Den haben wir nicht gesehen.
Ja.
War nicht dabei.
Den haben sie auch hingelegt, wo der
Okay, dann machen
Rockcracker
wir
ist.
das nicht. Dann machen wir sofort, wenn das vorbei ist.
Dann gehen wir saufen zu Dicker
Dann haben wir abfotografiert.
Kinderexperiment.
Kinderexperimente holen den Max, der darf dann entweder das zurückholen oder liegen lassen, wie er will. Das dauert nicht lang. Und dann
Also um halb 3 sind wir fertig mit dem, dann sind wir um halb 4 wahrscheinlich fertig.
wahrscheinlich früher sogar.
Ja, bis wir alles, ich muss schauen, ich muss die Dings noch aufholen. Ich muss das noch hochheben. Also
Ja, wir
um
machen
halb 3.
2,5 Stunden hast du das eingestellt gehabt, die Uhr jetzt. Ne?
Genau, um 2, oder musst du es noch zurückpumpen?
Ja, das weiß ich,
Und dann
ja.
ist es halb 3.
Okay,
Dann muss man die Box oben auffahren, dann runterfahren nach Dicker, dann ist
das
es
geht ja aus.
3, also später wie halb 4 wird es nicht.
Ja, genau. Weil wir den letzten Test machen oder was? Bist du nicht mehr? Du willst. Ja, sag ich nur.
Ist mir wurscht.
Also,
Also ich weiß jetzt nicht so gut, wo das liegt.
ja,
Was sind
ich
das für
kann das
Waypoints?
gerne, wenn du willst, draußen sein, dann was
Ja.
anderes machen, die holen, keine Ahnung, weil
Ja.
mit dem
Passt
sind wir
dann.
da wegfahren, allen die Waypoints.
Genau.
Also,
Und am Schluss, wenn du dann einfach dann übernimmst du noch und dann machst du nur, also die Suction Jar 1 und 2, die sind voll.
die 1 und 2 praktisch fein, zweimal. Ne?
Genau, die sind voll von der Experimental Side.
Also wir sind schon voll.
Die
jetzt.
sind voll, genau.
Wir sind voll, also wir können das Dreier nehmen.
Genau, das 3er, 4er, 5er, 6er. Und vielleicht auch feines und ein grobes und dann
Oder
gescheit Muscheln.
einmal fein, einmal voll.
Und dann gescheit Muscheln und Tube-Bombs wieder so raus. Und dann hat er
Schütteln oder was.
einfach so eine. Den Suction Sampler. Dann hat der Alessandro und der Ian haben dann wieder einen Bogen.
Ich weiß schon, was du meinst, ja. Also nicht Muscheln, nicht zusammen.
Nein, nicht Muscheln, sondern einfach nur
Gescheit einmal
Zeig.
hinfahren,
Ja,
ja.
gescheit einmal hinfahren.
Wie lange hast du das eingeschaltet gehabt gestern?
Viertelstunde. Viertelstunde.
Beides. Nein, insgesamt.
Gemeinsam, ja.
Gemeinsam.
Also 20 Viertelstunde bis 20 Minuten. Man sieht was reingeht. Und währenddessen lasse ich einfach kurz stoppen und umdrehen. Also und nächster Chart, was ich anfange. Das geht nicht schnell.
Weil das kein Problem ist. Also das können wir auf jeden Fall was machen.
Weil das dauert nicht lang und haben viele Leute was davon. Also 4, 5 haben mindestens was davon.
Ja, eigentlich sind nicht guten Sachen rausgekommen. 30 oder was? Oder?
das weiß ich jetzt nicht. Also meine Leute fixieren, wie gesagt, zum Fixieren die Sachen, weil sonst geht es ja nicht aus. Also ich tue einfach die Hilfen mal raus.
Dreißig, 14:15. Passt, weil heute ist ja
Good feeling? That thing tag over there?
Und schickst einfach an oben. Ist gut essen, weil ist ja wurscht, kannst du essen gehen.
Ich kann auch da bleiben, wenn du bist.
Dann komme ich runter, wenn ich fertig
dann
bin
bin ich
und dann
weg.
Ja,
Also du gehst zuerst, dann. Ich brauche nur sitzen.
Brauchst nur sitzen.
Nur sitzen.
Nur sitzen.
Okay, ich glaube, ich muss was noch abhaben.
Genau.
Kein Problem.
It's okay, so you know, just your copy. So you can between 12 and 2. Just to fix the soap dishes slightly in the shower.
No, because but I don't think it's not enough. It's not long. I don't use it.
All right, everyone. If anyone's wondering where we are in the world and where we're operating right now, we can just give you a little idea with some Google Earth here. So we are on the East Pacific Rise. As we zoom in here, you'll be able to see the track line that we took from Punta Arenas, Costa Rica. Took us about 6 days to get here, so quite a ways away. But the closest place to us is actually Mexico at the moment. But we'll zoom in here and give everyone an idea. So the bathymetry that we received from AUV Sentry from a previous expedition is what the researchers have been using to plan all of the dives and the locations that they want to go to. All of the GIS data or the geographic information is what helps us to plan the dives out in advance. And then if anyone was listening yesterday, we were mentioning the different positioning systems. So we have GNSS topside, which is like GPS, but also using Beidou and GLONASS, other constellations. And then we have the USBL system, so an acoustic positioning system with beacons on the ROV, so that when it's underwater, that's how we position it. So GPS doesn't work underwater. Obviously, if you need antennas are only going to work above the surface. So I can give another zoom out to kind of put it back into context for everyone, but the very high resolution data that we collected, or that was given to us from the previous expedition that was collected by AV Sentry, you can see compared to the background data that you can see in the blue there that Google Earth displays, and then when you zoom out even further, when stuff gets blurrier and blurrier, kind of off to the other areas here, those are unmapped, and that's only displaying satellite altimetry data, where satellites that fly overhead measure the kind of gravitational anomalies in the sea surface, because when there's a large enough mountain underwater, it will actually collect more water mass around it. So very helpful for us that we have kind of the preliminary data set from thousands and thousands of passes of satellites, and then we can go in with the acoustics, the multi-beam echo sounders, and map them to even higher resolution from the ship side, and then send an AUV down to get, again, even higher resolution closer to the seafloor. So most of everyone else has gone to lunch right now. But once we finish lunch, that's when we'll do the tour through the lab spaces with the scientists. Our chief scientist, Dr. Monica Bright, is in the room, but she's busy typing up a lot of notes and plans for the upcoming dives, which is a pleasure to work with her because she is organized, and if anything changes or something needs to be altered, we get plenty of heads up so that the ROV pilots and technicians can prepare the submarine for any new equipment or anything that might need to be going on there, and we just have the timing down pat. It's makes life a lot easier for the crew here on board Falcor. Yeah, big thumbs up from Monica. Thank you, Monica. And the pilot, who is seated right now, making sure that Sebastian doesn't float away from this box, is Chris Ingram. Chris, I can turn your mic up there. You want to say hello to anybody?
Hello, everybody.
There you go.
Yes.
Got them all. And then the co-pilot sitting here is Russ Cofield. How's it going, Russ? You want to shout out anybody? That's not that mic, it's this one.
Yeah, just say hello to mum and dad. They're sitting at home watching the video. So yeah, enjoy.
Well, I don't know that. That's a little specific to your parents.
Yeah. I'll save the shout outs for tomorrow. It's it'll be my dad's birthday tomorrow.
Okay.
And we had Yvonne's birthday yesterday, one of the scientists on board. So we got cake. I think there's another birthday coming up soon.
There were
No,
three.
Miro's at the end of the month.
Yeah.
Miro, our chief engineer. Miroslav Mirochev. And I don't think we'll have to go and find his passport from the purser's office and see just how old he's turning. I don't think he's going to tell us.
I wouldn't believe him if he did tell me, remember?
No. So we can do a little bit of recap of some of the stuff we shared yesterday. ROV Sebastian has the capability of going down to 4,500 meters, so 4.5 kilometers. We have about 6 kilometers of umbilical, or the tether, the cable, on a drum. So if we do need to do a retermination, then we can chop a little bit off, and then the slant range from where the vehicle is to the vessel, so Falcor 2 to Sebastian, ends up being longer. It's a diagonal, so it's not, we don't go straight down. We like to be out from the ship a little bit, but not too far. So right now I think we're stretched out about 80 meters, 81, yeah. So we're not too far away from Falcor 2. I guess it's the other way around. We're not too far from Sebastian. But we do have the extra cable on there. And Sebastian is powered through the umbilical, so we can stay down indefinitely. The fiber optics allow us for as close to near real-time response for the manipulator arms, 4K vision, and then all of the other video cameras that we have on Sebastian. We also have the suction sampler and capture jars for that. We have the CTD that Alex was mentioning earlier for conductivity, temperature, and density, which translates to salinity, temperature, and depth. And we can calculate the speed of sound from that. So as you go down through the water column, the speed of sound changes with the increase in temperature, speed of sound gets faster, increase in salinity, speed of sound gets faster, and increase in density, speed of sound gets faster. But obviously as we go down deeper, it gets colder, it gets warmer at the top, colder as we go down through, then we hit the thermocline. Then there's also the halocline for where the salt layer, and then the density increases steadily down to the sea floor. So with all the different changing variables, and you can see, yeah, so Chris is zooming in on the CTD there for us. It's kind of the thing that looks almost like a shotgun microphone from a camera. So all of the sensors are in there. There's a camera just to the left of it that we're covering up with the picture-in-picture. But using the sound velocity through the water column, we find out how fast the sound would be traveling through there. And then when we emit an acoustic signal to the beacon to interrogate it, we measure the amount of time between sending out that signal and getting the response back. So we know the speed, we know the time, and from that we can calculate the the distance. So, or the depth in this case. And then the angle of where it is, so we calculate the rho theta, the theta being the angle of the signal coming back to the USBL head, and USBL stands for ultra short baseline. So between the different transducers on there, we can determine what angle the signal responded from. What's for lunch?
Yeah, I had the sticky beef. It was really good.
All right, sounds good. So everyone out there, Russ has just gone off to get some lunch, and Carl Hill has come in as co-pilot. So we'll log a watch change here. And anything to do with the vehicle or science, we log in our logging software called C-Log. So right now we'll just do a pilot change so we can keep track of how long each individual pilot or tech is stationed in the respective roles here in the control room. And that's just kind of to track either development of new people like Tyler, who's an ROV pilot in training, currently serving as a 4th Marine technician on this expedition, and then just kind of make sure that people aren't hogging the stick too much. But yeah, so the logging software, every time that we take an event, sorry, you guys are probably hearing that loudly, I'll stop tapping the table. Every time that we take an event, all of the information that the ship and the vehicle are collecting is going through our influx database and OpenRV DAS to log all of the data. But at the moment that an event is taken, all of that data is captured, kind of like a screenshot of that point in time. And then we go back and we'll see actual frame grabs or real screenshots from all of the cameras that we have aboard ROV Sebastian, all of the sensors that we have, and then as well as all of the sensors ship-sided. So anytime we collect a sample or we spot a certain organism or geologic feature or anything, or take a water sample, we know where the vehicle is at that point in time, and then all the data that you guys see on the screen, plus quite a bit more data that isn't very useful to display, but the depth, the salinity, the temperature, the oxygen saturation that you guys can see on screen. And then another important thing is the time in UTC, so universal time. So everything on the ship runs in UTC, so that no matter how many time zones we pass or cross through, a lot of the times will be, right now we're staying on Costa Rican time, but technically I think we should be on Pacific time, Pacific Standard Time. So every time that something happens on the ship, we can reference it back to UTC. And then any other details that the visiting science party, the data loggers, will be logging. So either a species name or where the sample is located, where it was placed on Sebastian, whether it's in one of the bio boxes, the coral quivers, one of the push cores, or even some of the milk crates if we're just doing kind of rougher geologic collection of some big rocks, which you guys will probably see later. We're going to give a try. Yesterday we had a smaller pry bar, but the rock down here was so hard to break free, but then once it did, it was almost like Zach, the RV supervisor, mentioned it was like a St. Rupert's drop. So the kind of the glass that's dropped into cold water and is incredibly strong, but once it breaks, the entire thing shatters. So that's kind of what happened with the newly formed volcanic rock, and I won't speak too much about it because I was in the office doing some paperwork when that was all going on, so I kind of missed the interesting details of that. But speaking of Zach, your ears burning?
What?
Talking about the brittleness of the rock after we verified the break.
Yeah,
Prince Rupert's drops and all that?
Yeah.
that's the one. I was saying St. Rupert. Yeah, Prince Rupert drop.
We'll see. Victor, say the glass.
This guy out there.
Oh, what?
Mhm.
Not too sure. That's maximum already. Is the mic on?
It is. Yeah, I can turn it down. Zach's gonna open up his can of soda. There you go. Is that the non-caffeinated one? Just in it for the sugar, baby.
Caffeine after lunch will get in trouble.
Yeah.
Can't go to bed on time and cause a ruckus.
So there's a question here. How does the pilot log begin and end? Essentially, it's just whoever's on watch in which position. So there's the pilot, co-pilot, and the stream/logging station. There's also another marine technician station if there are any issues that are going on that we can hop in and sort. For all of the acoustic positioning, the inertial navigation system that's running on the vehicle, and then just the data integrity and logging of all of the video and the audio overlay as well. So to any grad students that are watching this in the next 5 years or something, I apologize for droning on about this stuff. This is all recorded to our DVRs. So we have redundant DVRs recording the signal coming up from the sub in case any of the files or the devices should become corrupted or faulty. And then we log that onto our NetApp array, which is actually a petabyte. So oodles of storage, kind of future-proofing for upgrading to 8K or higher cameras, or any other large data sets that we'll start collecting in the coming years. And so it's logged on the NetApp. We are also backing it up on a Google Transfer appliance, which we ship back to land, and that will be uploaded to the Google Cloud, or the bucket that we have, so that anyone anywhere in the world can reach out to our data architects and be able to download that data when it's available. It's one of the core principles of Schmidt Ocean Institute is open data sharing. And then all the software that we use, that we produced in-house or funded or contributed to, that is also available. We have a Schmidt Ocean GitHub that people can download from. And in mentioning that, OpenVDM, one of the developers, WebPinner, we work closely with him. Shout out to WebPinner. How you doing, buddy? And we use that program to scrape all of the acquisition computers, whether it be an EK80, which is kind of a qualitative single beam echo sounder with a bunch of different arrays and frequencies. We have the multi-beam echo sounders. We have the EM124, the EM712, the EM2040. Those are all Kongsberg made. We have our underway seawater sensors from the Seawater Lab that, if you were watching earlier, you would have seen with Alex Havens. So we have fluorometers, thermosalinographs, transmissometers, and pH sensors in there right now. We also have meteorological sensors. So we have PAR sensors, so photosynthetic active radiation. We have radiometers, which record long and short wave radiation. We have wind and MET sensors, so temperature, humidity, barometric pressure, wind direction and speed, and dew point. And then we also have a optical rain sensor that we have set up there. And then, am I missing anything else? Did that one. Oh, and then obviously the tracking of the vehicle, the CTD. The CTD that you saw in the bay, it was it had the caution tape around it and everything. If anyone was watching earlier, it's because we removed the Harken block that we have to pass that through to do some maintenance on that. The chief officer, Jason Garwood, was going around, and all of our lifting equipment gets inspected every 6 months. Make sure that it's in fine working order, and then tags of a certain color are placed on it, and the other one's removed, or anything that doesn't pass the test gets decommissioned. So that's for either lifting heavy equipment, lifting scientific equipment, any of the harnesses that we wear if we need to be climbing, going into enclosed spaces, down into tanks, up onto structures on the vessel, making sure that everything's in good working order. So we were just taking down the block, giving it a nice good clean and overhauling, and then it will be going back up. But the Niskin bottles and everything that you saw there when Alex was pointing them out, we have a suite of sensors on the bottom of that frame. So the bottles get triggered by the water sampler, and then underneath we have dissolved oxygen. Oh yeah, we also have a dissolved oxygen, or a oxygen sensor on the ROV, obviously because you're looking at that on the screen. We have dissolved oxygen sensors, conductivity, temperature, and depth on the CTD. We also have a fluorometer on there, a transmissometer, and a PAR sensor, so the photosynthetic active radiation sensor that I mentioned earlier that we have up on our MET station. What else do we have on there? That's it at the moment, but we also are able to put on any visiting science equipment on there. The mass spectrometer that was brought out for this expedition is still being worked on, and hopefully we'll be able to get that on for tomorrow's dive. Which reminds me, I'm going to go check in with Stefan a little bit. But that's enough rambling from me. Again, we're at the top of the hour, so you are watching live streaming video from ROV Sebastian, broadcast from research vessel Falkor 2 of the Schmidt Ocean Institute. I have been John Fulmer, the lead technician on board here, joined in the control room with Dr. Monica Bright, Zach Bright, no relation, Carl Hill, and Tyler, I can't remember your last name.
Take a while to make most common name.
You'll have to say that when you get on. But I'll hand you over to Tyler, you're in good hands.
Just call it Chuck Norris.
Yeah. Yeah, he looks like Chuck Norris. He didn't get on the, he was the one who was walking around with the camera with Alex earlier, so we're going to flip the script on him later and get him in front of the camera. He's got a real face for radio.
That's a face only a mother can love.
So he's a better looking Chuck Norris.
There's no better looking Chuck Norris. That was the fountain of knowledge, John Fulmer, exiting the control room slowly with a wave of the hand.
Probably still talking as he walks down the hall.
Quite a loquacious fellow.
Oh, I forgot like 4 more sensors he's thinking right now.
Yeah. Did he interrogate you about your ROV history, Joe?
No.
How did you get started in the ROV game, Carl?
You can smell the aqua there.
Uh-huh.
Well, so I joined the military out of high school, worked on helicopters. I did that as a civilian for a while as well. I had, I don't know, I think combined about 12, 13 years on helicopters. And one of the places that I was doing work was going to move locations, and I didn't like the new location where it was going to, so I started looking into other options. I looked into SpaceX at one time. It was kind of local in the area. There was pros and cons to that, but inevitably, like when I was doing research, I ended up finding a video, and I can't remember the company that did it, but it showed an RV video, subsea, obviously. And as soon as I saw that, I remember telling my wife at the time, that's what I want to do. I was so excited about it. And I applied for a couple different companies, and yeah, within, I would say, about a week and a half from seeing that video, I was actually an RV guy at that point. So yeah, I got hired on with Helix and worked in oil and gas for about 10 years, and that's when I heard about this and thought the science side of this would be entertaining and a different change of pace. So I came over here, and that's how I ended up doing this.
Yeah.
Yeah.
Carl and I have a slightly similar story. Slightly. I also saw videos and thought, man, that is a real cool job. How do I do that? And it was of Schmidt Ocean Institute and videos of Sebastian and the build particularly. But rather than doing it right away, I went back to school for a couple of years. So a bit of a delay there. But you've been with Schmidt for what, 7 months, Carl? 8 months?
Yeah, about 8 months.
And what's the coolest thing you've seen so far?
Man, you know, I think we did that octopus odyssey.
Yeah.
I've seen a lot of cool stuff, right? But what was really intense about that one was after doing ROVs for 10 years and seeing, you know, a few, maybe a small handful of octopus, and then suddenly seeing 10 times the amount of octopus I'd ever seen in one frame was pretty wild, pretty intense. So that was cool. We did see a shark yesterday. That was pretty awesome.
Yeah.
Those are kind of rare. I've only seen a few in the time that I've been doing this, and we got a really good clip of that oceanic white tip. It was really neat. So.
And if you're watching yesterday, that was right at the end of the dive when the ROV was almost being pulled out of the water by the A-frame. Caught a glimpse of the shark. Carl did in the bottom of the camera and said, "Hold on, we got to take a look at that." And so for the very limited amount of time and umbilical we had, got a quick shot of that. It's very cool. And I'm brand new to this, so every time we go down, it's amazing to see what's on the cameras. So it's also good to hear someone like Carl, who's seen a lot of stuff underwater, still constantly blown away by what we're coming across. These images from the bottom of the ocean are incredible, and oftentimes we're seeing things that no one has seen before, like last expedition. You know, we're going to places that had not been explored before. So the first people to lay eyes on an area is pretty humbling in a way, I'd say.
Yeah, definitely. Also, I really like the mid-water stuff. You know, we see a lot of things that I don't think you would ever see had we not developed technology to do like ROVs and this kind of stuff. Like a lot of this stuff is just not on the surface, and not only that, but then it's one more thing for me to learn and get better at. You know, I've always flown. Well, I've flown for a while now, but adding this high resolution camera and the more variety of lighting situations that we can create it's a lot of fun to like learn it and try and perfect it, and then still fly well to make a good shot. So it's a new level, or another level, I guess, of what I've been doing. So I appreciate that a lot. It's a lot of fun.
Yeah, keeping it fresh. And for someone coming in with not nearly as much experience as Carl, the amount of things you have to keep track of as a pilot while trying to get a shot that our outreach team can use for the videos, for social media, you got to track a lot of stuff in your head all at once.
Yeah, you kind of build like a mental image of your surroundings and where you are, because you can't always see everything, so you just have to just kind of know where it is. And, developing that sense really helps, so you don't have to think quite as much. You can focus more on the details of your shot. That's one thing, but that just comes with time.
Yeah. And when I've been in this chair, in the multimedia chair, I try to show multiple camera views so you can kind of get an idea of what the pilots are looking at. Because starting off fresh, that was an initial challenge, is to keep looking at all your cameras to see what's going on, not to get focused in on one camera, and to use multiple vantage points to understand where the ROV is in space, where the manipulator is in space. And you got to reprogram your brain just a little bit. Another cool thing about the midwater, as well as when you guys watch the live streams, often, we start on the bottom, so we're on a surface, and the surface of the ocean is another surface, but between those two surfaces, there's 2, 3, 4 kilometers of three-dimensional space that things live in. And that's hard to wrap your mind around sometimes, as people were very kind of on a plane, right? But these creatures live in a vast three-dimensional space. And it's pretty cool the things we've been seeing in there. What's been challenging about this expedition so far, using the manipulator, Joe? Or flying?
Yeah, the flying hasn't been too challenging. Definitely the manipulator work has been kind of on another level. It's just a very delicate process to get these boxes. Well, let's say the silicone, start there, like getting it in the perfect spot. It seems really easy probably, when you're just looking at one camera view, but it's so hard to tell the depth, like how far away you are. So getting the angles right, getting the silicone to land where you're being asked to land it is a little more difficult than probably it looks. Also the box, so we put that test frame, and that's really easy to like line up and tweak it until you get this perfect spot. But then when you try to replace it with the box, now the box is obscuring most of what you were looking at. So it's delicate, it's difficult. It's got that little gelatin barrier. Trying not to mess that up, and that just one little mistake could break something and cause this whole thing to have to start over or something like that, and that's stressful. So that's quite difficult. That's been the hard part, I think, about this one. The other two pilots that are off right now have been the ones that put the second box on, and I think that might be one of the most difficult parts, because one little twist of the wrist and you'll move that mesh box from below even a little bit, and it could compromise the whole test. So quite difficult to get that landed out without affecting things that you don't want to affect.
That's why often whoever's sitting in the multimedia chair, we tend to be pretty quiet when the pilots are doing those slightly nerve-wracking moves with the manipulator, because you have to be laser focused, and we don't want any distractions. And from my experience, I find it mentally tiring if you've been in the seat for a while doing a lot of manip work. I don't know, does that go away, Carl?
I think it gets better. I won't say it goes away. It is. There's a difference between obviously being physically tired and mentally tired. And if you spend too much time sitting behind these screens, depending on what you're doing, you will definitely feel that pretty quickly, how mentally tired you get. A lot of it is, trying to use two different images and create like a third, basically an image in your own mind of where you are in space. That takes a lot of mental, I guess energy is the right word, I don't know, mental focus to create that image. You're really not solely using one image to operate these arms and to put these boxes down, and it really takes a lot out of you. I went to Schilling years and years ago, and they had this experiment up, and it was a really cool way to kind of explain this. They had several cameras mounted up, and there was a desk full of like different colored bouncy balls, and they turned on one of the cameras, which is kind of a standard camera that we use for ROVs. It's not even as good as the ones you guys are seeing here, but they turned it on, turned the display on. They asked me how it looked. I said it looked great. They bounced the ball in front of it, and they asked me how it looked. I said, looks pretty good to me. And it did. I didn't see any blurring or anything. They're like, okay, leave that on. They turned on their new camera that they were working on, and they were using some kind of a video compression So they were able to get more frames and more detail out of the photo or out of the video with the same amount of with the same fiber that we're limited to get to the control room. And because they had more frames and whatnot, when they turned it on, you could suddenly see on the original lower grade camera the blocks that were being missing, the frames that are missing. So the ball would be, you know, in one location, and then the next frame it would be a couple inches higher, and so your brain is filling in that missing frames without you even knowing it, and so that causes a whole lot of fatigue. Well, the new camera filled that in for you, so it helps your fatigue. So I'm sure we're getting a huge advantage with some of these better cameras that we have on board here, but it's still doing it to some degree, I think. And depending on which camera you're looking at as well, it may be worse or better. And so yeah, your brain is using a whole lot more focus and energy than you even realize when you're dealing with what we're dealing with down here.
And I think even like grandmaster chess players, they've done experiments with them where there's physiological effects of being in such deep concentration for so long. Yeah, it is taxing in a lot of levels.
Yeah, you know, it was earlier today. I had one of the 7-function controllers, and even just trying to hold. We were doing the silicone.
Yeah.
Trying to hold this controller, which it's not heavy at all, but just trying to hold it very steady and not move it for, you know, periods of time, 10, 15 minutes, with very small changes. When we were done, I had to shake my hands out. Like, it actually hurts. It's like, why does this little light controller hurt? But it does when you're trying to be that focused with it, that detailed with it.
And that's why I like to be in the control room, because I can watch the different pilots and how they hold the controller. You know, what do they do? What's your strategy for opening and closing the jaws? Because it's the things you don't think about, how you're holding your arm. Are you using two hands or not? When are you indexing? What is the method for trying to get the most delicate grip on the jaws possible? It's all those little fine motor things that maybe they do subconsciously, but I'm consciously trying to pick up, how do you do that? How do you do that? Because it is. You don't want to screw anything up, and it's very delicate work. And, I mean, Lord forbid you sneeze or something while you're holding the arm, and all bets are off, right?
Oh, good Lord, yeah. Yeah, that's a good point. Just like when you're driving for very long, it's a lot like that, but it just seems like it comes much quicker when you're just dealing with TV screens.
Yeah. We are bathed in blue light here in the control room, aren't we?
Yeah.
But, I mean, you guys are rotating in and out and trying to keep fresh.
That helps a lot.
Go get a little sunlight, a little vitamin D, stretch your legs out.
Yep, yep.
After lunch, about 15 minutes.
Well, here on Falcor 2, we are cycling through our lunch period, so we got team members coming in and out of the mess. And then once our lunch is over, we'll be taking you back onto the ship, into the labs. We'll talk to some of this wonderful science team about experiments they're doing, how things are going, hopefully give you a little bit closer up view of what's happening in there rather than the sort of run through we did earlier today. So, yeah, and in the interim, we got a nice view of some tube worms. But we will keep you entertained here on the seafloor as we wait for this dye to set in experiment number 3.
Or if we kill the mowers, if we'd be better with all this.
And we do have a question about the mess.
Not much.
And we
Yeah,
eat the
earlier,
mess
it's just
and then come back.
schmutz.
think everyone needs a break from the control room. Everyone needs a mental break period, so the mess is a good spot. You're gonna have some nice windows out to the ocean. We do have monitors up in the mess, so you can keep an eye on the feed, which is always fun. See what the pilots are doing, see what's going on. But yeah, I think in general, people will go up, eat lunch, you have a little bit of social time, and then come back down in the control room because everyone needs that break. It also helps that we have some pretty good chefs on board, so you get a nice variety of stuff and pretty good meals every day. Looks like we may have spotted a, I'm not even going to take a guess. I am not a scientist. We do have some knowledgeable people on board.
Got some banding.
From the back, Salva has given us the definitive claim that this is indeed a fish.
That we was.
We just lost it. Kind of needed to see way out there.
Yeah, but I think maybe the shutter speed, sometimes that helps. Do 150. He's gone anyway.
Yeah.
Dang it.
We've seen that before. We could just fly and chase them, right? They wouldn't mind.
Yeah. How long is that too?
Just go. So, yeah, look right and up. I don't mind. Just see if we can see maximum banding. There's two kinds, I think. There's that kind that's digital, where it's like it's a hard line, and then there's the kind that the boomers do.
Yeah.
More like a gradual brightening and darkening. That's from the boomers. Shut that off. Oh no, it's still there.
Well, there's one on the screen.
Yeah, but they definitely do. They do that.
Yeah, yeah.
That's the boomers.
The boomers do that.
And that, I don't think you can get rid of that with the shutter speed. I wish you could. If we have mechanical shutter, you might be able to.
Just taking a moment here to flip through some camera settings.
What are you doing?
See if we can get the best picture possible. Pump out to you on YouTube and Facebook.
Did y'all go check out the kids experiment already today?
Oh, yeah.
See, he's got fur on his claws. See that?
You want to give me a flag average on it?
You got it. So we got another few minutes before we take you back out onto Falcor 2 into the lab spaces. If you guys have any questions, feel free to send those in the chat. We will do the best to answer them as we can. Cool.
They want to see the bunks we stay in.
Bunks look like bunks.
Yeah.
Yeah, that was it. Sweet.
Do you want me to keep flagging this or you get it flowing?
No, it's good. Yeah, I'll just do it along with this.
That's good. The long pause here on the seafloor does give us time to peer into the lives of the organisms down here and get some nice close-up shots. And I do have to constantly remind myself that if the RV wasn't here, this would be pitch black darkness. Are those the Alvin weights there? Yeah. Also seeing some evidence of previous expeditions. This is a well-documented, well-explored site. And members of our own science team have been here, 4 or 5 times since 2003, I think. Perhaps even more. And we do have a question on Facebook. Are there any scientists from the earlier dives that are still on board and able to see the progression of exploration in their experiments?
Oh, that like dropped off these experiments here? Or like the weights and stuff?
Yeah, that's a good question. If that question is about previous experiments in this area, I can't answer that.
Nah, everyone's at lunch right now.
Yeah. And we have a pretty bare control room right now because everyone's enjoying the concoctions of our chefs.
You can see some of cubes from Monica.
Oh, they're Monica's the ones?
So, some of them, I believe they were from Monica's past attempts. And she was deploying also those sponges with Sabine. This that we are seeing for trapping the small animals, the meiofauna.
Okay.
And I think when you deploy, you are relying on projects. So you have a project for deploying your stuff, and you might count on being invited for next cruises and be able to recover. But I think this is not always the case. And also the pandemics was disrupting all the science. So not every time that you deploy your experiments you can get them on schedule. And I remember that Monica was commenting that she was losing a lot of deployment because of the volcanic eruption of 2006 in this area.
Oh.
That's a good point, yeah. And introduce yourself, Salva.
Yeah, I'm Salvador Espada. I'm PhD student of the University of Vienna with Monica, right? And this is the second time I come here. I was here in 2009, and we went down with the Alvin, and this is a very interesting experience with the ROV.
You went down with Alvin, so you were in a manned sub here?
Yeah.
Wow.
That's cool.
Yes.
So you actually physically been here?
Yeah.
That's interesting.
Crazy.
How different, Salva, is it being on Alvin versus seeing it on these screens? What's the experience like?
It's very different. I was given a camera to hold in one hand, and I had one little window with this circular shape, and I was the scientific observer of, I think, the port side, and I had Monica in the starboard side as scientific observer, and then the pilot. and I would have the control of one camera, one outer camera, and this I could operate, and I had something to write and record the observations, and this was my mission. I was scientific observer, and we were told to identify the bucket leads because they were not much sure of the exact position. So I had to really pay attention through the window if I identified, and then one I spotted, and then I told the pilot, and then we managed to get to the intended recovery. And all what I'm seeing here, this was 2009, so now it's 14 years later. So especially this navigation model of the terrain, the bathymetry, this has progressed a lot.
Yeah.
How long were you in the sub for at a time?
I think deck to deck it was kind of 8 hours.
8 hours?
Yeah.
It's a long haul. So when we talk about scientists having been here before, we have scientists on the ship who have literally been to this site in a manned submersible. They've seen it with their own eyes.
Yes.
Human operated vehicle.
Yeah, all the information that was hanging there, it was big red marker of the heading on top, and that was it. So it was much different.
Remember the drawing? I'll just make something chocolate black and I'll change it if it's not.
we've got just a few minutes before we will do our more in-depth tour of Falcor 2's lab spaces and talk to some of the science party. Alex and John are getting that all set up. And so we'll just do a quick recap of what's been going on here so far today. So right now we are 2.5 kilometers below the surface of the Pacific Ocean. We are on the Underworld of Hydrothermal Vents expedition, and you are looking at live images from the seafloor via ROV Sebastian, the remotely operated vehicle that's operated by the Schmidt Ocean Institute. My name is Tyler Smith. On this expedition I am a marine tech, and in the control room right now we have Alex Paris, pilot, we have Carl Hill, co-pilot. We got one scientist in the back. Salva's back there. It's a pretty bare control room at the moment. We are wrapping up our lunchtime here on Falcor 2, and currently we're in the midst of an experiment on this expedition. So in our picture-in-picture you can see two boxes. One box is a vivid crimson red, and the other box is a less red. There we go, some deft camera work by Chill. And these two boxes are experiments where we are pumping dye into the boxes, pumping the dye back out eventually. But the scientists are staining everything in that space. And then we're going to remove the dye, remove that outer box, and we'll come back later. And if we see any organisms that have moved into that space that are not dyed, we'll know that they have reached that area, not through the water column, but perhaps through the seafloor itself. So the theory we're working with here, the idea the scientists have, is a novel one, that organisms can travel through the hydrothermal vents, so beneath the surface of the seafloor, to repopulate areas. And so that second box, you can see an example on the right side, is just a mesh container. So that prevents organisms from getting in through the water column. So if they are in that box and they are not stained red, we know they've reached that area by other means. And so it's a lengthy experiment on these days. It starts off yesterday actually with the science team selecting a site they think would be ideal that has some hydrothermal flow, and sometimes they'll seal off parts of that crack and they'll get the area prepped, and then on a day like today we'll go down. The pilots will put some adhesive caulking on the seafloor and set down a series of these two boxes. Now the mesh box has a very soft, is it silicone on the base, Salva?
I think so, yeah.
And so that will, with the adhesive caulking, stick to the seafloor with hopefully no gaps. The second box is laid on top. It's a very delicate process, and then the science team will pump that dye into the box to coat everything. Now it is non-toxic biodegradable dye, it's been well tested, so it achieves its purpose with no harm to the organisms or the area.
It is good to have 3 replicates, we call them replicates, because you make a scientific observation once, and it might be you have good luck and you saw something, and then you think it's what happens every time. But if you see 3 times, then you can be more positive that this is a real phenomenon. So it's very advisable to repeat the observation at least 3 times, and then that gives you more confidence once you see that they all agree, which is not always the case, but we hope to be lucky, and for sure we try the replication.
Yeah, so three different experiments that are replicating. And if you've been tuning in from the beginning, I think the team has gotten a little more efficient in putting these boxes down and getting everything going, because this is the first time these have been deployed on the seafloor with an ROV. So the team tested them previously in 3 or 4 meters of water, right, with scuba team, I think. And so the science team and the pilots, everyone is working on the best way to deploy these experiments, and they are not easy to get nailed down 100%. But you guys seem pretty happy, right?
Yeah,
The deployments have gone well.
We are super happy. There's many things that can go wrong with the pumps, with the holding, with the sealing, and there have been a big effort of development, and I'm very impressed on how everything is flowing so smoothly. I think Monica and Sabine can be very proud of the work that they are leading, and the people in the Slovenian Institute of Biology were very important on the development, and in the Royal Institute of Oceanographic Investigation in Netherlands, and in the University of Vienna. And this is a big progress of the Monica Bright long tradition of underwater deployments.
And that brings up a good point. So we do have, it's a really international team, right? So scientists from Austria, Slovenia. Where are you from, Salva?
I am from Spain originally. I'm from the south in Andalusia.
Yeah. We've got a Costa Rican student. We have Americans. So it's a good international crew on here.
One guy from Brazil.
Right? Yeah. Yeah. France.
Yeah.
Germany too.
Yeah.
Yeah.
And one Italian student.
An Italian. Yeah. Yeah. So we got a few continents represented here on Falcor 2 today with Science Party. The crew of the ship itself is also very international. Philippines, Ireland, UK, America, New Zealand, South Africa. So yeah, a lot of nationalities represented on this one ship out in the middle of the ocean. What are these guys, Salva? Do you know?
I heard that those are serpulid polychaetes. I'm not so much of a zoologist myself. I'm doing my PhD on bacteria, but I think I heard that these are called serpulids. Maybe somebody in the chat can correct me. So these are polychaetes. I think they are related to the big ones, to the riftia. So these feathers you see, the plume, would be the equivalent to the red thing we see in riftia.
Oh, cool.
Like how they catch the gases and exchange the gases with the water column, like how they breathe. And in this case, I guess feel the feed also.
So those feather-like structures are similar to, on the larger tube worms, the red plumes. So they
Yes.
operate similarly.
Yeah.
Very cool.
So,
And these white filaments are bacteria, sulfur-oxidizing bacteria. These, I think, are probably related to the Brigatoa that were the first found at the 19th century. The end of the 19th century, they were the first that were found in the rivers to make the chemosynthesis to oxidize the sulfide. So where already very long ago they were finding that there were some life based not only on sun energy but on chemistry. So these are the first discovered chemosynthetic organisms. And this white color of the some bacteria organism, this is indicating of usage of sulfur, because they take the sulfide and convert it in elemental sulfur that gives that white color. So many of the symbiotic organisms also look white because of this elemental sulfur storage.
All right, and our team of Alex and John are. Let me hit my mic.
The white is elemental sulfur that is some bacteria use it as a storage intermediate product. The sulfide is the most energetic compound, which it has 2 atoms of hydrogen, is 1 atom of sulfur, SH2, and it can be oxidized, obtaining energy. And an intermediate product is elemental sulfur that has 2 less electrons, so it typically becomes sulfur globules in the bacterial cells, and this gives the white color.
Right, excellent. So you're going to some in-depth explanation of what's going on in this frame, and that is a unique benefit to working with Schmidt Ocean Institute and having access to control room. It's like a one-on-one science lesson every day you're in here. And soon we'll be taking you in the lab and give you a little more science knowledge. So John and Alex are getting mic'd up. They're getting set to take us live into the lab. We'll talk to some scientists, and we'll get that going here in just a minute. So until that, enjoy some more close-up images of mussels and riftia. We have our 4K camera. All right, the moment has arrived. We are going to briefly leave the seafloor in our dye box, and we'll be headed into the main lab to talk to some scientists. So we'll send you over there right now. Enjoy this break and a little more look into life and science on Falcor 2.
Okay, my name is Ingrid. I'm part of the team of Monica Bright, and now I hear myself double. I'm very sorry. That's okay. I'm here to make release experiments with the muscle Bathymodeolus thermophilus, which is this muscle you have seen on many of the live videos and on YouTube. And this muscle has in its gill symbionts, which are chemoautotrophic, and the muscle feeds on the leaking secrets which coming out of the cells and on the bacterial cells itself. What we don't know is what happens to the bacteria after the muscle is dying. Can they escape from the tissue, or are they doomed to die with the host? So what we do here now is we cut out a little bit of the gill, and then we put them in a little capsule which shows Teresa.
Hello, I'm Teresa. I'm from the University of Vienna, and I also work with Monica Bright on these ELISE experiments, and this is the capsules. So we put tissue inside the capsule and close it and put it into this pressure chamber.
So
this.
If the bacteria can escape from the gill, then they go into the water, and then we offer them, because they love to settle on surfaces, on super tiny little cover slips, which Teresa prepared right now. And for that, I think you have to come really close because they are tiny.
So I stuff these cover slips into this rubber, and then afterwards in Vienna we make fluorescence in situ hybridization.
Yeah, and this goes in also. Then we fill it up with the seawater we filtered from down, and then we just close it. And then with this pump we can add a pressure of 220 bar, 3300 C, which is adequate to the pressure down at the hot vents. And then we incubate it. After a day or 2 days or 5 days, of course, because we don't know how long it takes them to get out, we open it, and then we fix the samples for different analysis, which will be done in Vienna. Then if we find them on the cover slips, we know they can escape from the gills, which would be an advantage, because the larvae don't have the symbiont. They have to find the symbiont in the water or on the biofilms. And it's good if they seed it out again. When the host dies, they get out into the water and it gets seeded. And this is what I have to do here. And they say, well, thank you very much.
So you've been doing this then for each of the experiments that are up here?
Yeah. Yeah.
How many? Let me put myself on. All right. So now I'm going to ask a question here. How many mussels then have we gone through with this on this cruise?
Until now, I did 8 mussels.
Wow. Okay.
But I have to go on again with 8. So I will do 16 experiments before we go back to Panama. Yeah, this is it. And Teresa, we go on now with the giant tube worm.
Yeah, and for me, basically, it's the very same setup, and it worked for us. So we could, with fluorescence in situ hybridization, show that Endorhiftia is escaping this little capsule where the tissue is inside and settles on the cover slips. And now we want to see that when they escaped, if they can also switch from the chemoautotrophic metabolism to the heterotrophic metabolism. So what I do is I feed the escaped symbionts with glucose and glycine, so sugars and amino acids, and then we check for the metabolism, we check with fluorescence in situ hybridization if more escape, and also check with transcriptomics which genes are upregulated.
What pressure then are we pumping that container down to?
Yeah, we try to go as close as possible to the pressure what is in situ.
Okay, I'm just going to show
Yeah.
how thick the acrylic is there then. And is that pumping done here then with the?
This is the pump, right?
All right.
So we pump it here, we attach it, we open it, and then afterwards we close it, but we put it in the fridge at 4 degrees Celsius to have more or less the degree Celsius that is also down there.
Excellent. Well, I think we'll sit back on over to Tyler back in Mission Control, and we'll get a couple more scientists mic'd up. But thank you so much for showing us the work you're doing here.
My pleasure.
And see you, but my pleasure also.
All right, so we got a nice glimpse into some ongoing experiments in the main lab of Falcor 2. Now our team's going to set up, I believe, with another group of scientists, and so we can tackle another experiment, give you another peek inside what's happening with the science party and in the main lab. And again, when they reference Riftia, these are these tube worms you see in this shot. So the white tube and the red plume sticking out, this is what they are studying. So we on Falcor 2 are definitely happy that we have this technology, that we can take you into the labs when we have these pauses and a dive such as this, to give you a better idea of what the ship looks like, what the spaces look like, what the scientists are working on. It gives you a deeper dive, so to speak, into this expedition. So thank you for joining us, and we'll be back here momentarily with another glimpse inside the labs of Falcor 2. And right now, Looks like there's a lot of plumes out. We've been watching these dive streams. Often when Sebastian sets down, you see everything get retracted. It's an immediate action or reaction to the motion down there on the seafloor. But these creatures look pretty relaxed, and maybe we can find an eel pout, which is maybe the most relaxed fish that I've seen. Yeah, there's a distinct eel pout deficiency in our frame right now, huh?
They have found new locations to be lazy.
Yeah. They can be lazy anywhere in the ocean. It looks like we can see a little bit of the shimmer at the top of that tube worm clump. And again, that shimmer does represent the hydrothermal flow with these vents. So that water we've measured, I think, up to about 20 degrees C, so much warmer than the ambient 2 degrees C of the seawater surrounding it. And often it will be mineral rich, so different maybe chemical composition. Alright, we just gotten word from one Alex Havens that the next interview segment is ready to go in the main lab. So I'm going to send you over there and we can learn a little bit more about the science from the scientists.
All right, there we go. We got a lot of channels on our communication here. So last time you heard about the pressure chambers, so we do have quite a few in our fridge here. And you can see that these fridges get packed pretty full. This is just a 4 degrees C fridge, but we also have freezers that run all the way down this way. And if you were around for our tour earlier, there were others as well. So we're going to come on over to two more of the scientists here, have them introduce themselves and kind of what they're doing. A little bit different layout here. So take it away.
So, hello, my name is Finalizing. I'm from the University of Hamburg. Currently, I'm working here with people from the NEOS from the Netherlands, so the Royal Institute there. And I am currently sorting very small animals out of a sample that we got from yesterday evening. We have to do this quite fast because these animals die rather quickly, and we need them alive for the experiments in the pressure vessel. We also keep the samples here on ice in small petri dishes or small glass wells, and we also have one of the wells which we don't know if they're alive yet or if they're still alive, so we're going to sort through that afterwards still again. Yeah, and we sort out small crustaceans currently because these are quite sturdy, so these samples are from 2.5 kilometers of depth, so they come up a long way, and they, if they want to be alive, the animals have to be quite sturdy. And the small copepods, that's what the crustaceans are called, we put them out, put them in these glass vials, and then I just hand them over to my colleague Alessandro, who is doing the experiments.
Hello, I'm Alessandro. I'm currently doing an internship at NEOS with Sabine Goldner. And yeah, basically, I'm continuing to pick all the copepods from these samples. And it can be quite difficult actually to find out if they're alive or dead, depending on how long they've been stored in the fridge. Sometimes you have to use these really small, basically like toothpicks almost, I don't know how to call them, and really gently touch them to see if they move. It's quite tedious work. This is an example of one of the samples. And actually, I don't know if you can see them on camera, but these really tiny red dots are the copepods. So yeah, they're much easier to see than shallow water copepods, which are generally like quite transparent or gray. Then what I do is I use the exact same pressure vessels as my colleagues, and basically pressurize them using this high pressure liquid chromatography machine, and then incubate them in this water bath. So you can see inside of here the two pressure vessels currently at 22 degrees for a 8-hour incubation. And basically, once the incubations are over, I take them out, I check all the parameters I have to check, and especially how many are alive or dead. This way we can find out, even in the long term, how much, theoretically, they could survive for their travel in the subsurface.
All right, so I have a quick question then here. Curious, obviously the pressure, big thing, and you're repressurizing them, but how many would you say are surviving? Like if you get a sample, are there just one or two that you're having to find out of those samples, or, I mean, are a lot of them moving usually?
It really depends on the sample. So especially how well insulated the sample is. So for example, in some cases we had some like errors where the samples were taken up and the water was too hot and most of the animals were dead. After some troubleshooting, we've managed to get way more of these small animals alive. And for example, yesterday, the vastest majority were. So yeah, it really depends on the conditions where they come up.
With the microscopes, there's a lot in these little dishes. How many would you think you've found at this point? How many have you processed?
Oh,
if
hundreds.
you had to guess, hundreds. Okay.
Hundreds, yes.
All right. Well, I think we'll toss it back to Tyler once more in Mission Control and see if we can wire up any more of our scientists here to go over their work. But thank you so much.
Thank you.
Thank you.
All right. So another visit to the scientists, Fanny and Alessandro, explaining some of what they're doing aboard Falcor 2 in the main lab. And it sounds like Alex is going to try to set up another interview in the labs. Until then, we will have the very soothing images of the seafloor 2.5 kilometers below the surface of the Pacific Ocean. These are live images coming to you from ROV Sebastian, the remotely operated vehicle from the Schmidt Ocean Institute, and it is tethered by an umbilical to research vessel Falcor 2, which is what you just had a brief glimpse into the main lab of. So again, those two boxes are ongoing experiments for this expedition, those dye boxes, and this is the Underworld of Hydrothermal Vents expedition, and we are on the East Pacific Rise.
Yes, much more life. the Mid-Atlantic Ridge. Yeah, there was, you almost couldn't see it. It was so dense with critters that you just a blanket. It was crazy. A lot of different features with where the venting site has been. It made these great big giant columns. They're very brittle. This is more hard, like rock. So quite different structure as well. Yeah, it was very different. Very different.
So that was one of the pilots, Chill. He was on the Lost Cities expedition on the Mid-Atlantic Ridge, right?
Yeah. Mid-Atlantic Ridge, yeah. We were just talking about the differences between these two different sites. Just from a visual perspective, it was overflowing with these shrimp creatures. They're solid white, though. They're not the red ones that you think of. And very dense population of them. Also, the formations that occur from the venting create this real crumbly, kind of like rock, but it's really delicate. Which, in contrast, over here we don't see quite as much of that. We see it a little bit, but yeah, we're much smaller towers here. Maybe that's just because it's a newer site. I'm not really sure, but the ones here are like white. The other ones, Mid-Atlantic Ridge, were these brown kind of structures. Just vastly different, completely different.
Yeah, there's various types of hydrothermal vents, right? So you have black smokers; those are the very superheated ones. You can have white smokers as well. I think the Galapagos has both of those types. And then there's the Lost Cities type of vent, which I think discovered in 2000, 2001. So a different type of vent with different chemistry, different structures. But these vents have only been known to man since what, late 70s? 77?
I think the 60s, actually the Cold War was a huge driver of the research of the bottom of the ocean because of these military submersibles launching nuclear missiles. So they would want to find the places to hide and how to be able to reach the coast of the other block. So there were also a race, same time that there were the race to the space and the moon, there were also a race to the bottom of the ocean with all this military situation, and it was also a time of scientific revolution concerning the geology with the plate tectonics, proving like this was proposed 50 years before, at the beginning of the 20th century, by actually I think it was an Austrian geologist, Wegener, that he realized that the coasts of Brazil would fit in the Gulf of Guinea, and he was positing the hypothesis of the drift of the continent, and it had to be middle of after the Second World War that they found this proof right with the magnetic anomaly of the spreading centers. It's a very interesting scientific story.
Yeah, fascinating. Changing scientific thought with these discoveries. It's incredible stuff. And I've just gotten word from our Intrepid crew in the main lab that they are ready for another look at the science with the scientists. So without further ado, we will send you over to the main lab and see who Alex has decided to talk with next. Here we go.
Hello, welcome everyone for joining us. My name is Jan Steger. I'm from the University of Vienna and was invited by Monica Bright. However, I'm working at the Department of Paleontology, so not the Marine Biology Department, and I'm focusing on the mollusks from the vents, which we get up here with our ROV Sebastian. And as you can see, we have already gotten quite a lot of different samples of many species which are really cool and in a very good condition. I'm just about to catalog them to get an idea and good overview of what we have, and also not to lose any of the precious data. And my research is about studying not only the living mollusks, but also if you can find any empty shells in this environment, which is quite cool, as paleontologists study a lot, of course, empty shells and fossilizable hard parts. But at hydrothermal vents, it's pretty difficult because the environment is dominated by those hydrothermal fluids, which are quite acidic and tend to dissolve a lot of shells. However, no one has really studied how much can be preserved and for how long, and this is actually what I'm really interested in. And therefore, I'm going to collect as many different species as possible. And what I'll also do is to have a look at the shell structure, so the microstructure that you can only see under a microscope. I may show you an example here. So, although the shells of mollusks are made of limestone, so basically calcium carbonate, it's not comparable to anything you would see in your kitchen pot, for example, when you heat water. And this is because the mollusks have special proteins that help precipitate the mineral in different shapes, which gives them very particular mechanical properties. So the shells would be very strong and efficient armors to protect them from predation, but also from the chemical environment, which is, as I said, quite acidic. And depending on the species, we can see very different structures. For example, those here are two examples. At above, you see two images of the shell of a single species, but they have different layers, and you can clearly see this in the pictures. And each of them has different mechanical properties that, for example, prevent the spread of cracks when there's a predation event. So not the whole shell will shatter, but the break is basically stopped. And I'm going to prepare sections of those shells that we collect here to look at them under the electron microscope and find out more about those particular structures and how they're related to the different groups the mollusks belong to, or if there are any adaptations to the specific vent environment.
All right, and a question that we have real quick that I have seen asked quite a few times when we're looking at these mussels up close. We are frequently seeing, it looks almost like ash on them, like these little gray flakes. What is that on the mussels?
Yeah, actually we have an example of this here. Right, these are the vent mussels we see quite a lot during the Sebastian dives. These are pretty small still. And on the surface you see what we call byssus threads. So these are threads made of protein that are produced by the bivalve to anchor themselves to the substrate, and also used in forming these dense muscle aggregations. However, the mussels are not fully sessile. That means if they don't like a place, they are also able to change their position by abandoning those threads and attaching themselves somewhere else where they find more suitable conditions. And the threads that they had previously remain actually stuck to the substrates, which are often other shells of mussels, and they consist of a small string, which is quite strong and has really is a very powerful means to attach to the bottom. But they also have a pad, which is like this flaky thing that we saw on the shells, which is very characteristic and stays in place even a long time after the mussel has moved away.
Fascinating. All right, well, thank you so much for sharing your work. It sounds like you're going to have a lot of work when you get back to shore, too.
For sure, yeah. And it's really good we have. I mean, getting samples is not easy from the deep sea, and we are in a really privileged situation with Sebastian to get so nice material regularly on a daily basis.
Excellent. Well, I'll toss it on back to Mission Control there with Tyler. And I think we've got a few more folks that will mic up here. Thank you so much.
Thank you.
All right. Nice scientific interlude with Jan there. And we're back with our dye boxes on the seafloor. But a benefit of these trips into the main lab, not only do we get to see the scientists, but we get to see the domain of the marine technicians a little bit more. So you guys are familiar with the ROV team and what they do. You're watching it right now. You know, we're seeing the output of that. You've heard the wonderfully informative Alex Havens on the stream quite a bit, our multimedia technician. And the marine techs work often behind the scenes, so they're managing the lab spaces, helping the scientists, tracking and maintaining the sensors aboard here. You heard John earlier list off a quite voluminous amount of sensors and pieces of technology on Falcor 2 that group of crew is responsible for. So they do a lot of the work that allows navigation to work, the USBL, a lot of the systems that we rely on here with ROV Sebastian. The marine techs do a lot of that behind the scenes stuff. So getting a glimpse into maybe the parts of the ships that they work more often in is also a nice look at how Falcor 2 functions. So here we are. Another look at the boxes. Has anything changed? No.
Zero thing.
Pretty close to the way we left them. No eel pouts. But a beautiful view of the seafloor nonetheless.
Could be one right there. That one's been there a long time. So
Oh,
it must
yeah.
be another level of lazy, if that is one.
The first eel pout we're looking at. Since the dawn of time, he's been there. Do you guys recall when our 3 hours started?
Yeah. Oh, look at that.
Oh, ask and you shall receive. Oh, he's frisky.
I have it here, 17:26 UTC.
Almost 2 hours.
One more hour to go. So the suction sampler energy of suctioning, this is not a big deal. Like the consumption of the engine, is a big part of the consumption.
No. And the suction sampler runs off a hydraulic motor that runs off the main. So it runs in the same circuit as, say, the thrusters and the manipulators. And when you're using the suction sampler, typically you're not using a lot of other hydraulic power. So it adds a minimal amount of extra load on the system, but at a time when you're not really consuming that much.
So this is
anyways.
It's not a significant consumption.
No. The biggest consumption for power on a ROV is almost always the propulsion. With this ROV, you can quite easily use all the horsepower, and we do when you're just flying it. That's your biggest power command. When you're sitting down with a little bit of trims on, it's actually not much.
And is it true that the vessel's engines are electrical also?
Yep. So most boats of this class, like workboats, they'll have a series of generators that will be used to power usually a pair of common buses, like power buses, that feed electric thrusters. And so they'll run at, try to keep it like a somewhat optimal load range for the motors, and then the ship itself can actually turn on more or less generators as the power demand changes. So this boat has 6 generators, and they can, we're actually running, depending on weather, run on 1 or 2 while we're sitting in the station.
And is there a conventional engine as a backup, or?
Yeah, so they're, oh, for propulsion? No, all the propulsion is this electric, yes, all diesel electric.
Okay.
Yeah. So the thrusters in the, after the vessel, are the thrusters used for propulsion are the same ones that are used for dynamic positioning.
Yeah.
In the bow of the vessel, there's two tubes that are perpendicular to the vessel, as well as one retracting azimuthine thruster. Right now, they're only using one of the aft thrusters and the tubes in the bow to keep this in place.
All right, I've just gotten word that Alex has managed to grab a couple more scientists, so we will see what's happening in the main lab once again, and we'll send it over to Alex right now.
Hey, my name is Tinkara and I'm from National Institute of Biology, Slovenia, and here is my colleague.
I'm Nicole, I'm a student at the University of Vienna.
Here we study viruses. In fact, we try to understand how many bacteria from the deep sea are infected by viruses. Viruses are very small, but very diverse in their abundance and in their reproduction. And not unlike other organisms that actually produce their food and energy, viruses also rely on their host for reproduction. So in order to reproduce, they actually have to meet their host. In the ocean, the most abundant organisms are bacteria and archaea. So that means that basically is the host that virus most likely encounters. So actually viruses that are infecting bacteria are also most abundant in the ocean. And here we are talking like 10 to the 10 per milliliter surface water or 100,000 per milliliter in the deep sea. So by doing an experiment here, which is called like virus dilution experiment, we study viral production. We actually concentrate a community of microbes that presumably contains also bacteria that are infected with viruses, and then we isolate them in small vials, and we observe in time viral lysis of the host. And when this happens, the virus is released from the host and the viral abundance increases. So we are going to count the samples later back in Vienna. While by lysing the host, then we have a release of the dissolved organic material and nutrients in the ocean, and by this, basically, viruses are importantly contributing to the cycling of organic carbon in the ocean, and also by just controlling dominant host populations in the environment. And in this way also allowing more rare taxa to increase. And now Nicole can explain what we are doing here with this complicated filtration system.
So usually what we do is we get a sample and we filter it first with a big filter around 3 microns, and then we move to the smaller filters, which are these two. And in the end, we end up with a concentrate that has a lot of bacteria in it, and we dilute it with the virus-free sample, and then we incubate them in these pressure chambers here. And every 6 hours, we have to take a sample out and fix it so that it can be measured back in Vienna how many viruses are in that sample.
And so with the pressure chambers, right, you said you have to open them up pretty frequently. How long does it take to pressure it back down and do that back up? How long are you spending just opening and closing that?
It takes a while because we have to really screw these screws on very tightly. Mm-hmm. But actually pressurizing it doesn't take so long, around 3 minutes or something. Mm. Yeah.
And then I saw that you all were just running something here, but obviously the ROV is still down in the water. So is this some sort of cleaning? Are you preparing for the samples to come up?
Yeah, we are testing some new tubes because this pump works in the way it's a peristaltic pump, and it's continuously pressing a lot on the tube. So with time, we run 5 experiments up till now. it gets bruises. So we need to replace the system. As we work with bacteria and viruses, we really need to make sure everything is clean and sterile for the next setup. And actually now Nicole will soon sample as well. So.
And so if you're preparing for the ROV to come up, when the ROV comes up, is it kind of just like a rush? I mean, is it exciting just waiting there for it to show up and you're like kind of stretching, you're getting limbered up, or?
It's definitely a time when you know where to find all the scientists on board. We are like everyone with his own like bottle and vessel, like a Christmas is coming, you know, like yeah. And then we are running, yeah.
Well, thank you so much, and I think we'll toss it on back over to Tyler in Mission Control and see what's going on the bottom of the sea, but thank you again.
Thank you.
So,
as Alex requested, we're going to see what's happening on the bottom of the sea. And I don't think much has changed since the last time we tuned in. Looks pretty similar.
Still 2 boxes.
Still 2 boxes. But definitely a big thanks to the scientists so far who have participated in these little interview segments with Alex, giving us their well-spoken descriptions of their experiments and giving us an inside look at what they do. And we may have some more scientists later, but we'll keep you informed if that happens. But as of right now, we are just peering at this box and the tube worms, the mussels, some crabs here on the seafloor 2.5 kilometers beneath the surface of the East Pacific Ocean. Alright, so we've pulled in a little bit of the bathymetric data that we're using here on this expedition. So we've had a close-up view of the area where the boxes have been deployed. But if we had unlimited visibility, and well, the seafloor doesn't look that color, but this is what the bathymetric relief looks like of this area in the East Pacific rise. So in the center of the screen, you can see a little bit. That is Sebastian. The line trailing down is actually following Sebastian into this landscape. So right now we're in a little bit of a canyon feature, but we're very pleased with this high-resolution bathymetric data that was provided before this expedition, and it gives you a great look at the dynamic landscape that this expedition is tackling right now. So the past few days we spent a lot of time on the seafloor running experiments. This gives us a nice overview of what the surrounding landscape would look like. I've got a question, Sal. So right here, there's a lot of life. How far away from the vents before the life sort of falls away until it's much more sparse?
The rest of access is a desert. So you have some small animals, but very sparse populated, and some other flying fish. I think we will have some cruises of access, some dives of access in the next days, and we also want to have a look to that environment. And is really all this life which you see here. We will see that is not more there. And this is the big extension of the bottom of the ocean is really a desert of life.
Excellent. So apparently, according to Saba, will be off-axis. So we will be able to see the comparison between the density of life at these sites, what you see on the camera right now, versus how desert-like it will become pretty quickly. So the area we're in right now is really an oasis of life on the seafloor, and it's a wonderful view. But I have heard from Alex that we have some more scientists lined up, so we're going to kick it back over to the main lab and give you an in-depth look at a little bit more science.
Hello, I'm Avery Fulford. I'm from Seattle, Washington, and I'm currently living in Cambridge, Massachusetts as a first-year PhD student at the MIT and Woods Hole Oceanographic Institution joint program. Here I am working, we're basically the prokaryote team, so that's both bacteria and archaea, so some of the simplest but coolest, in our opinions, forms of life. What I'm working on specifically is basically every time we go down to the seafloor, I take a rock sample, and I'm really interested in looking at the basically the activity of the microbes living both on the surface of the rock and basically potentially inside the rock too. So I actually have here a rock that I can show you guys. This was collected in the sampling that we did yesterday. This is from Fava Flow, and it was basically taken just from the seafloor. And yesterday we actually took off some filamentous sulfur oxidizing bacteria. So they form these little chains that sort of make the rock look hairy almost. And we took those to sample to test, you know, sort of their metabolic activity and to take a closer look at.
Yeah, so Avery is a hard rock scientist. And I'm Clarissa. I'm a postdoc at Woods Hole Oceanographic Institution. So we work in the same place. And whenever you see on the screens or on your screen at home, Sebastian going through the water column, you might see things that look a bit like snowflakes appearing in front of the camera. And we actually call this marine snow because it looks so much like a snowflake. And marine snow is biological material that is sinking through the water column. You can imagine it a bit like a compost pile. So in your garden, you have plants growing, and these plants, when they die, you put them on the compost pile and they decompose. But the ocean doesn't have a compost pile. So the biological material that grows at the sea surface or in the deep sea will just float around and be decomposed there by bacteria. And I study what the bacteria do with that biological material and where the marine snow goes. And so I have my corner over here. Marine snow might look quite large on your screen, but actually it's like something that you can barely see, like maximum a millimeter maybe. So I typically try to transfer it into very small cups or into the petri dish here with the pipette. And then I take microscopic pictures of it. I'm not sure if you can see it here, but if I... Like here you can see a marine snow particle on the camera. Let's zoom in a bit there. This is an example of what is actually floating in front of your camera. And so bacteria that find that marine snow, they are in paradise because they found a huge, huge cake to live on and feed on as much as they can. And while they do that, they do a lot of different things. That includes respiring oxygen, but also transforming other elements. And I built an incubator to simulate natural conditions for the cake and the bacteria to study how much oxygen they consume and how much other processes they do. And since it's so cold down in the deep ocean, I'm going to take you to the cold room to show you the incubator. And so
In this room we have 5.5 degrees Celsius, so it's almost as cold as in the deep sea. And since it's also dark there, I'm keeping the incubator under a blanket, so they are not disturbed by light. And you can see a lot of different little bottles here. Each of these bottles contains one marine snowflake, hopefully colonized with lots of little bacteria. And you can see the pink spots in the bottles. These spots quench light depending on the oxygen concentration. And this is the sensor. It flashes in green. And the green flash basically triggers the spot to quench the light. And the light fiber cable will also read how much the light was quenched. And in my computer here, I will get information on the temperature and on the oxygen in the glass. And so over time I can see how much the bacteria have been breathing. And afterwards, I will take out the marine snow and look at it in various ways to see what it's composed of, what kind of bacteria were actually in there. And so it's the first time I'm ever looking at marine snow from the deep sea. So I'm very excited to see what's in there at all and what's happening at all. It's a big surprise for me.
I guess that kind of brings in my question, and I'll have you both there then. So you mentioned first time looking at it from the deep sea. What would your normal, like, how does this differ from what you're used to, I guess? I mean, obviously you're working in a big fridge. Do you normally work in a refrigerator, or?
So my last particles were from Bermuda. That was quite warm, so I could keep them at room temperature, which was nice. And if you're like in the middle of the water column, it's like 12 degrees Celsius, so a bit cooler. And the other big difference is the pressure. That's something I cannot simulate here, of course. So, yeah, that might be a problem, but yeah, we're getting as close as we can.
We've seen some people using like the pressure chambers.
Yeah.
You've got your incubator. That probably doesn't pressurize as nicely then.
I haven't managed a way to pressurize it yet.
And also, you know, I had a question. With the rocks, right? Like we're seeing them collecting these rocks. Do you are you watching the screens going like, oh, that's the good rock, get that rock? Like how into the rock collection do you get then for the ROVs?
Oh yeah, definitely. I mean, we're hoping to have more rocks with sort of a biofilm on it, so that's like this really, thick conglomeration of a lot of different bacteria sort of forming colonies on the rock. So when you have sort of this more densely populated rock, that'll be better for my analysis. So definitely we watch the dive stream basically from the conference room right next door to mission control all day, and try to look at the rocks that we're getting back up on the surface later that evening.
That's a question. The question I had there was about how much were they watching the screens going, oh, grab that rock there, grab that one. So, all right, well, we'll back on out of the cold lab here, but thank you all so much for showing us your work, and I'll toss it back to Tyler.
Thank you. Bye.
And thank you again to Alex and the scientists for giving us a look inside their scientific experiments here on Falcor 2. And also gives you, viewers, a good idea of where these samples are going, the spaces they're going into, the scientists who are handling them. So when we pick up something from the seafloor, who's looking at that, who's using it, what science are they doing with those samples. So it gives you a little bit better look at the whole process of what happens. Because Alex has pointed out a couple times, when Sebastian comes back to the surface and is set back on its cradle, and the ROV team gives the scientists the go, it is an orderly but a mad dash to get those samples off the sub and into the labs as quickly as possible so they can start their work. And there is often a tension of excitement in the air when the sub comes up, especially when some sought after or some really good samples are coming up. And you can see the excitement in the scientists just when Alex is talking to them. They love talking about their work, they love what they're doing, and we're privileged in this dive to have time to take you behind the scenes. And so now we've got a nice view of some stuff floating in the water column. A lot of soothing images here today and a lengthy experiment on the seafloor.
Players
And you were saying, so the particles like this with bacteria, are they throughout the water column typically?
Yeah. Wow. Yeah. Different amounts. I think here it has a bigger concentration because of the whole situation going on.
Wow.
For
That's super
example,
interesting.
iron is released at hydrothermal vents, like not here because it's rather deep, but in other places they found out that the iron from the vents stimulates primary production in the surface waters. Because the ocean is iron limited.
Yeah.
And phytoplankton needs iron to basically be active.
Oh, very
so the vents also have a really important cycle on the global iron and also so it's so tiny places, you know, but they're so important for the ocean.
Yeah.
And this we're just starting to understand at this moment. It's like,
The iron that is in the chlorophyll center, it's the iron is what it makes possible the converting the energy of the sunlight in bringing it to the building the chemistry.
So the iron is
the chlorophyll has one single atom of iron that goes from 2 plus to from 3 plus to 2 plus with one single photon that hits. It allows converting one electron in a non-energetic level, and this electron is taken by a chain to bring it to binding one hydrogen to a carbon. So this is how life molecules are built. And they say it was invented here.
This is we don't know.
So it's actually quite important that the iron makes its way into midwater, otherwise into the shallow waters, otherwise you wouldn't have photosynthesis.
Yeah, iron is also coming from other places, so it's like not only so it's estimated that these places make about like 10% typical cycle, so it's like not the majority.
Oh,
but
so it's coming from like rivers, from erosion? From like
yes,
erosion.
yeah.
Wow.
Yeah.
But like the whole water is basically a cycle through the Earth's crust and basically comes back again, you know, on the at these places here.
Oh.
Which is crazy, I think. It's like, you know, like for the entire ocean, I forgot like the number, but something like, I don't know, I have like something like 10,000 years in my mind, but I'm really not sure if this is true. That like it takes until the entire ocean water is like once like pumped through basically through the Earth's crust. You can get rich in all the chemicals. It's
That's
crazy.
incredible. So that makes a lot more sense. What you're, like I was trying to connect the dots with what you said earlier about like why it was important to study this, especially since people are looking at exploiting areas like this.
Yeah.
And it's kind of ironic because a lot of the exploitation is for like batteries and solar panels. For like no climate, but not actually know what damage you're doing. That's crazy.
So that's right now, for example, I'm working like on a paper, like also in a different political channels to bring this, you know, message basically to the people who are like in the city. Develop also like what area needs to be protected, and then like also this project plays into a role because, you know, if we can also show that larvae travel through this system, it's also one other, you know, indication how important it is to not only like protect a small space but like make it broader, basically. To protect like all the different ecosystem services that you know, the vents provide.
That's awesome. That's really good. I'm glad you're doing that.
You know, this looks like really like, you know, really freaky, but it has like a, when you look at it from the broader angle, it's
that's cool.
Yeah.
Gosh, that's so interesting.
It's amazing the impact that microorganisms have on ecosystems.
Yeah.
They're so small, but there's so many of them.
Yeah.
And so like, because they're small, it's easy to like completely dismiss them. But it's just like mind-blowing to think. Like the interactions between like the geochemistry and then like
It's all completely interwoven.
That's so cool.
Yeah.
You got it. I will segue to you. Now, you said we're high pressure man on the aft deck. All right. It looks like Alex has got another interlude for us, and we are going to be going onto the aft deck for the high pressure van. So, previous dive I mentioned that the massive aft deck of Falcor 2 means we can load different containers on there. So there is a lab space that has been put on the aft deck of Falcor 2. I have not been in there yet, so this will be brand new for me, but we'll send you over there for another glimpse of the science that's happening on Falcor 2 in the Underworld of Hydrothermal Vents expedition.
All right, so I'm Ian Hughes, and this is Dr. Jessica Mitchell, and we're aboard the Falcor 2. And we are from the Gurgis Lab at Harvard, and we're really excited to show you a little bit about our work here, which takes place primarily in this shipping container right here, using a system that was devised by our advisor and sort of updated throughout generations of students, and we use it to study deep sea organisms. So we can take you inside and give you a bit of a tour. All right, so what we're looking at here is the high pressure respirometry system. And so there's a lot going on here. It's really kind of complicated, but it has one simple goal. And what we're trying to do here is we are trying to study deep sea hydrothermal vent animals, and we're really trying to keep them alive and study them in sort of in situ conditions. So we're trying to replicate the kinds of water and gases and pH that they have in deep sea vent communities, but we're trying to do it aboard the ship, so we can study really what's going on and their physiology and all kinds of different aspects of these animals, and in many cases their microbial symbiosis. So there's sort of a lot of different kind of parts to this whole system, but a lot of it comes down to first we really have to make hydrothermal fluids. We have to like simulate exactly what these fluids are, and there's really nasty stuff in them. They're pretty low pH. They're sometimes they can be warm, but it's more diffuse flow, and there's all kinds of stuff like sulfide, gross materials in them. And we get our water from that we use on the ship. We get it from the surface, and so it takes a lot to actually turn surface water into hydrothermal vent fluid water, as you might imagine. So most of that takes place here in our gas equilibration columns. And so what we do is we pump in gases from gas tanks that are outside, and using these buckets or these boxes over here, we put very, very specific amounts of different gases into each column, and it's bubbled through. And so we have, you can see it's a little bit messy, we've been rigging them up, but we have four different gases here. And so our gases are red is sulfide, yellow is CO2, and then we have air and nitrogen. And so we can use these to adjust how much sulfide we have, adjust how much our air quantities, and adjust our pH and DIC with CO2. And they are then bubbled through each of these columns here using these air stones. And so we continuously have water basically being pumped in using these solenoid valves here, and gas is bubbled up to the bottom. And so what goes in is water and the gases, and what comes out is basically simulated hydrothermal vent fluid. And so, of course, we have to go from this fluid here that's just normal, like sitting in the surface, it's normal pressures, and we have to get it to really, really, really high pressures. And so we do that using these HPLC pumps. So we have, sorry, that's our reefer unit. But we have a whole set of high pressure gases. It will go off. Awesome. Yeah, so that loud noise in the background is actually a reefer unit, and even though we look relatively warm, it's actually 10 degrees Celsius right here in the van. So it's actually really, really cold, and then we usually have bundles of jackets that we have to put in every time we want to do research in here, and we spend hours and hours a day here. It's always weird going from inside the van to the nice tropical 9 degrees north in the East Pacific. But basically, we're keeping everything cold, and we have our columns here. Our column feeds to these HPLC pumps, and they go from our fluid, which is at normal atmospheric pressure, all the way to really, really, really high pressure. And in addition, we have a second feed with a second set of pumps where we can pump in things like isotopically labeled carbon or nitrogen, so we can do lots of really cool experiments. And from here, I'll pass it on to Jesse, who is going to show you really where the fun happens in our vessels. Sure.
Yeah, so I work on Riftia tube worms. So those are the big, beautiful tube worms that look like roses at the hydrothermal vents, and they really need to be pressurized to keep them alive and at hydrothermal vent conditions. And I study the bacteria inside them that cannot be cultured outside of the tube worm. So really, the tube worm for me is like they're a chemostat, a vehicle to keep them alive. So I incubate the tube worms under varying conditions and then look at the metabolism, the metabolic changes of the symbiont under these varying conditions. And so we bring them up. They get briefly depressurized before they go into the pressure vessels, and then as long as we get them pressurized again, I've got 4 worms in here and 4 worms in here, and you can see they're both at pressure, and fluid is flowing in to the tanks and flowing out of the tanks. And the beauty of this system is that we can test what's coming in and we can test what's going out. So I can measure how much oxygen they're consuming, and I can measure how much sulfide they're consuming through the symbiont's autotrophic pathways. Yeah, and in a few days, I'll be able to open the vessels and take the samples.
All right, so that's one of the questions that a lot of people have actually had, that is, we're bringing these critters up, can they be kept alive? And yes, they can, but like, what's the time frame? Like, how much of a rush is it when the ROV comes up, you're grabbing them? Like, how long could they live at 1 atmosphere? And then how long do you think you could have them in these containers?
So I haven't ever really pushed the limit on the containers before. My advisor says quite a while, but the cruises are short, and for my experimental purposes, I don't actually need to keep them under experimental conditions for that long. And as far as depressurization goes, it really depends on the vent organism. Riftia is one of the more delicate animals, I would say, and you want to put them back under pressure within a few hours, and the colder they are while they're at atmospheric pressure, the better they do, because it slows down their metabolism. And one of the things that they're pressure sensitive with is the function of their enzymes and membranes. So as long as we get them pressurized again within a few hours and they kept cold, they do pretty well.
All right, and then obviously you were talking about making hydrothermal vents, basically. Like you're trying to simulate the conditions they're in. So when you're watching the video streams, I mean, I think maybe you have the best idea of what it would be, right? Like it smells a little bit in here. You got the gases you're adding to the water. It's loud. Like obviously we don't want to put you under the pressure, but like you're just getting this thing screaming at you all day. So do you like look at the rift and you're like, can you picture it? Do you feel like when you see the footage, you're like, oh, like I know how that smells? Do you sort of like have that take the job home sort of thing now?
Yeah.
Well, a lot of people as well, like there's experiments going on right now with some food that's made. I suspect you probably wouldn't either.
Actually like the smell of sulfide, weirdly enough. And the weird thing about the vent environment is actually the conditions change really quickly, and it can be a little bit stochastic for a lot of the animals. So yeah. Because they don't exist in hydrothermal vent fluid that's coming out. It's actually a mixture of the bottom water and the hydrothermal vent fluid.
Yeah.
Because hydrothermal vent fluid is 400 C. So because it's a mixture, depending on the ratio of that mixed fluid, they can go from 3 pH to 7 pH within a few seconds, and the temperature swings can be really extreme. Oxygen, sulfide levels, the whole 9 yards. So it can be pretty chaotic down there, and I don't know, Ian, it's pretty chaotic in here too, right?
Yeah, I was gonna say, I'm like, we don't have the physical pressure, but sometimes, things go wrong, and we have a little bit of the kind of emotional pressure. So I think we're capturing most of the hydrothermal vent.
And I had one last question for you then. Like with all this making the water, how much of it is like, is it like a science of like, oh, just a little more, or are you basing this off of like solid values, or how much of it is tinkering, right?
It's 100% tinkering. Like I would say there is a very small portion of it that is actually like, it is science, and we know like adding more CO2 usually does drop the pH and things. But in the end, the first week we were out here, all we were doing is just equilibrating these columns. It was like, how do we get the sulfide to be just right and the CO2 to be just right and all these sorts of things. So it is, it's a lot of plug and play and just testing with the little droppers, exactly like a science experiment.
Thank you so much for giving us a little glimpse into the hydrothermal vent we've got back here, I guess. I did not, you were like, Alex, it's going to be loud when we go in there. And I didn't know quite what you meant, but yeah, it's quite loud. Our audience apparently didn't hear that much of it. They were able to hear you over it, so that's all right. But usually hearing protection in here, I assume.
Yep.
Yeah. Well, thank you so much. I'm going to toss it back to Tyler in Mission Control. But thanks for letting us see your work, and good luck out here.
Absolutely.
All right, another fascinating glimpse into actually lab space I have not been into. So we've seen a lot from the scientists. Thank you to all the scientists for sharing their work with us here on the live stream. And we're back. That was a fascinating glimpse at the lab on the aft deck creating hydrothermal vent conditions. There's been a little bit of action here. So rather than just the soothing scenes from the seafloor, the team has turned the pump on, right?
Yeah, we turned the pump on, and it is now removing the dyed fluid from the box and returning it back to the bag on Sebastian.
So that bag that is popping up on your screen here, you may have seen this throughout the past few dives. So we've used it to collect seawater. We also use it to carry the dye down to the seafloor, pump it into the dye box, and now we're pumping that out of the dye box. So this is a process that takes about 35 minutes. Empty that box out, and then the pilots get the unenviable task of carefully pulling that top dye box off. So a little bit more time here on the seafloor until we can get to that point. And in the control room right now we have Dr. Sabine Gallner, and we have Alex Parrish, pilot, Carl Hill is co-pilot, working the manipulator, and I am Tyler Smith. On this cruise I am a marine tech, and I'm filling in for the inimitable Alex Havens, who is taking you guys into the lab spaces and chatting with the scientists. As I look in Sabina, can you tell if anything's happening?
So everything is working as it should. You can also see that the bag is expanding. But what is different in this experiment than for the other two is that I think we have like more vent flow coming from below, which kind of caused that the stain was pushed out quicker than for the others. So this is something we cannot change, and it's there. It's, so we'll have to see afterwards very good how strong the staining is.
Interesting, yeah.
So this is what I'm currently noting down, what we write down. That when we recover this box in the end, that we carefully look how good the staining is.
Actually, because usually it's a lot brighter red at
So
this
at the
point
other
still.
dives you could see it like stayed longer in the box, and I think it has to do, and you can also see it like this area is more active. Also when you measured the temperature, it was higher. So it makes sense that the stain is getting out quicker. I just hope it's not too quick. It should be fine because we used a lot of this dye, so
Yeah.
it should be fine. But it's just something for us to write now down that we can check when we recover the stuff.
Did you do any testing for how long it takes the dye to affect these animals that you're trying to dye?
Yes, so about a year ago we did like intensive testing, like with also other marine organisms, and we tried different concentrations, different time lengths, and we basically overdo it a lot here. Like usually like just probably like
Right, so
a
we're
third
doing-
of the time would be enough, and like a concentration that's basically much less than we use.
So we're doing 3 hours, but you found that roughly 1 hour was probably enough. Huh?
Like we use here even a higher concentration than we used like for the shallow water experiments. So basically, like the amount of color that we use is much higher.
Much higher.
Yeah. Just to be sure that everything is stained.
Sure.
Yeah. So, but this we have to like really like basically like see when we bring it up.
So another good reason to have those 3 experiments, as
Yes.
I was talking about before. So this one, a little bit different conditions, the dye maybe not in the box as much, but you have the other 2 to compare it against. So
Yeah, so I'm not worried at this moment because also you could see in the beginning that the dye like entered in well, so I'm not concerned at the moment, but this is something just to watch out basically. And as you said, like it's really important, but in science in general, to have like more than one of these experiments because the different environmental conditions, there are many things that can go wrong or that you cannot foresee, like this case, for example. And if you only have one shot, then it's very likely that you miss. So, but like to find out basically what we want to study, one box would actually be enough. But because we know that things can go wrong, especially if we do it the first time, we always make like more. Yeah.
absolutely, yeah. Build in redundancy so we make sure we get what we want. What time did you get Kyle's chair, Alex?
Yeah, 12. Is that a problem?
No, I just got aquatic side, making sure you get part of that. For sure. I know you know, in your part you have. But for the water. So we have an interesting question, Sabina, about how did you guys design these boxes? How did you choose the materials? What was that engineering process like?
Yes, this is the first time that such an experiment is done in this deep sea, and we basically started from scratch. So basically, first the idea was born, and then we kind of put our heads together and thought about how we, how we can study and unravel the question that we had. So if the larvae can colonize the vents through the subsurface channels. So first it's about, okay, we use a box, the mesh size, this we knew pretty soon because there's like a kind of a standard like this 32 microns, we know like these limits. And then it's basically, we thought about like how large or small they should be. And then also work together a lot with so at the NEOS, the Netherlands Institute for Sea Research, we also have a workshop there. So also technicians that help us design all this equipment, that they can also build it for us. And basically what we do, we sit a lot together and think about solutions, how to put this experience together. So first to this mesh box and the staining box on top, it was actually the first design was already, we were already happy. But for example with the silicone sealing that you can see on the bottom, originally we had like a kind of a curtain that would extend, and like then with an iron chain below it with small balls of iron that would seal it to the box. But then during some first shallow water experiments 1 year ago, we saw that this is not working, so it's not sealing good enough. And then we went really into like testing materials, like we wanted to have something like between solid and sticky, and like solid and fluid and at the same time sticky. And then basically looked into what kind of silicones are on the market, and it was trial and error, and placing things in the in the fridge for like also for days and in seawater to simulate what would happen here. And then we found, okay, this is the material that we want to have. It was just, we just made like simple, like when we put the silicone together, we made like plates from 10 by 10 centimeters. The next step was to upscale it like that it would fit the box. And then also we had to think about a way how to mount this silicone onto the box. So then like we kind of, whilst pouring the silicone together before it hardens, we also would need to embed like a kind of a net that is then holding, that you can screw then onto this box. So it looks very simple, but actually until all these small little details are like done, it takes quite some time. And also just, trying out things.
Yeah, it's like most things we're seeing on these dives. It looks simple maybe, but there's a lot of forethought and engineering that goes into it.
Yeah, like from an outsider, if I would see it like before, it's a box,
Right, yeah.
Nothing special about it, but it actually takes quite some steps to come to such a design.
Yeah, absolutely. Is it performing as you predicted?
It's working really well, I think, at the moment. So we still have to, it's not done yet. So, but the first things that we see are like really good, and also really amazed how well the operation goes, because I've worked quite a lot also with ROVs, and it's incredibly difficult to deploy these things. It's like there are so many details you need to watch out, and it's such fine movements that the pilots have to do that it's really I think it's like they're so high skilled to put these things down. It's like I'm really like amazed by that, to be honest. And so it's like really working out really well.
just this shot we have up on screen is representative of countless hours of experience in engineering and research and coordination and timing and planning, and it looks easy, but nothing we do down here is easy. Very cool. Alright, I'm going to hop off the mic for a little bit, but Tomer, another one of the Marine techs on the expedition, is going to hop in my spot. So again, we are looking at live footage 2.5 kilometers below the surface of the ocean on the Underworld of Hydrothermal Vents expedition here on the East Pacific Rise.
ROV bridge.
Go ahead, Bridge.
Very much. I would just like to reposition a little bit. I was wondering if we could carefully move the waypoint and a comfortable spot west of you guys and hold the wind and the DP currents from the north. So I would like to get my head a bit more around the north.
Roger that. So you want a waypoint due west of the ROV?
Yeah, that would be possible.
We already moved it. 80. You should be pretty comfortable making a straight move.
Yeah, I think so.
I'll keep an eye on it.
One of the big things would be just watching over these cameras in case like
He starts pulling us.
yeah.
So watch your sit or something fixed.
I would yeah. I'll watch that. If you start seeing any movement,
I'll stop.
well, thrust down, like try to
Yeah, and then call him. Or just pay out on the way.
yeah, I mean we have options. I don't know if we need to stop it, but we can just manage it ourselves.
Yeah,
I
just,
think.
so basically just hold tight. Yeah.
On that same note about the boxes, I don't know who you guys communicated with as far as like the handles and like how you made this. Whoever it was, they did a great job with.
Yeah, so we our first design was a bit, so we had originally like 2 handles on, but then like we had got information back to make on the mesh box like this rope. Then we drilled the holes, but still like took the handles with us because I think I don't know like we just have like also several options basically.
Sure.
Because you don't know the setup like before, and we also like if everything would fit on the porch. We also talked about that. But in the end, it turned out different, and I think like very good, like how it's now everything is like fitting on the raft.
Yeah.
And also with the but yeah, but it also helps that if you've worked a bit with rafts, so you also know a bit like how things
Oh, okay.
where to.
So you had some experience.
Yeah. So not with
sandals are perfect.
Yeah,
They're good.
yeah.
Like you said, even the size of the box. I mean, if it was half-inch bigger, it probably wouldn't fit in our porch.
Yeah. So this we knew before, yeah. So we kind of also like knew how much space we approximately have.
Sure.
And Box, das Ding hast wie früher ausgesucht über die anderen. Ich muss mal schauen, ob der kurz das Ding sind. Das wird mit uns sehen. Ja. Die war die höchste Temperatur. Genau, richtig. Genau, schießen lässt, dann bitte. Nicht nur uns. Wenn es Sven macht, ist das eigentlich sehr wurscht. Wir müssen nur schauen, ob wir das austrainieren. Wo bist du gerade? Schauen wir, ob wir das Jahr haben. Auf Stunde? Weiter geht's nicht. Aha. Okay. Das ist einfach so nicht das ist einfach so nicht, oder? Nein, das muss nicht sein, das kann ein kleines Bildungshaus sein. Das ist halt so interaktiv. Aber schon. Ich glaube, die sind Babys, weil da mitten was.
They're moving the vessel.
Oh,
They
yeah.
move right now.
They want to face more north, so look at this.
Alright, I don't think they're gonna.
Yeah, it looks like we're doing this. So he's currently eagle-eyeing in case he starts pulling on us or something.
Yeah.
So. Offset.
Ram.
Yeah, I think we cut them custom code, and they were probably like starting to poke out a little bit now, but yeah, they're definitely the ones that we pulled back.
So, our big vessel moves, so we're just making sure that this.
Yeah, thanks. I felt the movement. I was like, oh, off the drive.
They're gonna move. I think it pretty much should be on.
Cool. Oh, somebody left the torch.
She's about the green one.
Yeah. How many? 11 minutes. 11
That was
minutes.
like biting into the dick. It's horrible. How long we got? 10 minutes.
I think the phone is really not good. It's like all out focus. Huh? So when you send me the pictures, I should get the sign but on which did you send the picture?
Inside the library.
Okay, but if you go on previous.
I don't know how to pronounce.
Signal, I call it signal, but I call it signal.
You know that.
It is on the focus. But it doesn't matter when you take a lot of pictures with your cell phone, they are out of focus. And you have to take the camera and learn to use
But
it.
I think this will be the camera.
Not just like, yeah, but
This is a front and it's not bad quality. I could recover
but
the
Salva.
better quality from the device, but I'm having difficulty from the device to the computer.
which device?
The picture,
Yeah, but then
I'm
I
having
should show you
No,
the
but
reason was
I
why you use
have a difficulty with this operating system of this computer. If you want that picture, it could be retrieved using your computer. The good quality.
Oh, well, it's a good quality. I see it's out of focus.
The focus is flat.
It's out of focus, then I don't
And
need it.
so you don't want this one. Okay.
So you will select some. And switch computer to like this.
With the camera.
This is better. Yeah, I'm going to go back that is a minute. So, and lift the picture.
I don't know if that is the. You should open the file to see the size of the file, to see what it will get. Otherwise, I have it here. I can email it to you.
Lift the picture underwater lifted. I have this inside.
So I email that to you both, right? You want me to do the picture?
Mm-hmm.
This is the one you want, right? Yeah, yeah.
I have this. I have this. Forget about it. I have it.
Do you have to what? The signal, it can be that it does not give it to you, it would require.
How should I know this?
And the size of fiber. The file size of fiber, there's it's not required.
I don't know.
If you open fiber, if you see the size of the fiber, you see it do not require.
Yeah, okay.
This is you know what you have. So is this the one you want? So you want this one you want? And this one you want? Yes, another one. That's supposed to be the same shape. I either say no. Which is this one. This one. Another thing. And this I said you also. I don't know if that's in terms of like traffic jam and all those things of the city of 2016.
So in 3 minutes we can switch off the power.
Okay. Yeah.
3 minutes now. It's like CT switch off. CT stays in your it's very big. It's for 100,000. But if you go like first one, it's like flow through. Yeah. Yeah, just like that.
Did you check on?
What was absolutely historical studies there's something people because destroyed because it's like the morphic journal facing a little bit some data that it's so far away from the down. Yeah, there's almost complete engines. Yes.
He's waking up.
It's a very strong material.
That's good. Because I've seen him in little and I'm like, oh, I'm taking some of that.
Yeah, that was the
Definitely clears the nostrils. He's like,
yes.
Breathe again.
Yes.
What is this like? Should it be integrated? How much what is the thing now?
CC dash B Y dash NC dash SK.
And then my name?
Yes, space 4.0. No, that's not like that.
Oh, this is mine. So this is for the, this is mine. Okay, I can switch off the
Alright,
So I make a biological observation or a frame
observation.
biological observation. That dye is to go pop off.
Yeah.
Did you send it already?
I sent you the 3 emails. I don't know how that will happen because the emails are sent. If you need them faster, I can also read internet.
Fix that.
Yeah.
I don't know what would do it.
I
I have no time anymore.
I didn't stay.
Where did you send it?
I sent you one picture twice and then if there is nothing, I sent you before.
I did nothing. So, okay, I'll do it later.
Go down and sit from this.
Say again?
Go down to sit from this.
Certainly. So I thought you were using that one.
I can't see that far. I can see it, but
It's not, no, it's just useless.
I didn't tell you that the backups are going smoothly. The backups
Yeah.
are running smoothly. Without the videos around 500 gigs, so everybody will be enough everyone drive.
Yeah, please.
Yeah.
Do you need the experiment switch? No, leave that one.
Only when we're on our way.
Oh, right.
Because of this. So now we carefully recover the staining box. Recover the staining box. Yeah. Okay.
Do you want to try and just look straight upwards do the same thing before using the, that seemed to work. Just using the rubber inside.
you are not getting the emails?
What did you say?
The emails came out of my email. So they are going here and then
Put it on the stick. Because I have the time to send it off, I need to send it off to the newspaper.
What's up? I just went in it, I just zoomed in it. So
enough view. Good stuff.
Just drop it in here, drop it inside. There's nothing in here.
Zoom out while I touch. Okay? Yep. Just give you an overall view. Some slope. Right, you want to pitch it to aft a little bit. And you need to pitch towards yourself.
You have to, otherwise it's opening up on the other side.
I think they again needs to come that way. Oh yeah.
Watch out, watch out. Slow. Otherwise the box is like removed from the bottom. Can you grab the yellow rope instead to lift it in addition? Because I think like... Oh, sorry.
Put it back down when I grab it. Okay.
Or if you...
Let's try and do as little movement as possible.
Yeah.
Just let that go now. This the jaw, to be honest. Not even so.
system, the Schmidt Ocean Institute, no?
You will not be able to send the 3 pictures in one go because it's beyond 10 megabytes.
Yeah. You zoom out a little bit. Good. It's easy.
The separation is still the same. The separation is the same, Schmidt Ocean Institute.
The legal frame is CCY, non-commercial share alike for
Same, very good. What it says for us.
Yes, this was.
We'll kind of move over to the side and then we can re-grab it. It's almost a little bit tricky. We designed an easier one. I can fly back if you want. Okay, I'm going to fly back. It's good. Fly back.
How are you going?
Well, I'm just getting my elevation, you know.
Like this, no?
What did you
I get settled.
CCY? Yes.
You got to watch out. Okay. So that's super exciting. Okay, just stay with.
It means awesome. Oh, yeah. Anyway, this is the way we set and give each other pictures.
That's
Yeah, just go from there. Or talk analysis, then we put in photos because there's also then we talk analysis pictures. Okay. Is it described the picture means?
there is no figure.
Yeah, you need to check that this is the picture is described and not only made it. But also needs to write in
I will include
the
the
text. This, okay. But people should also know what it is. Huh?
some of the pictures have the text.
Yeah, if it's let's say that if it's obvious, then it's very hard. If it's not obvious, then you have to think that people have seen this.
Have a butch further out. It's fully out.
nice statement here. Differences in the reef there. I got in. There's no attribution. This is mine. For me, or whoever takes it, I didn't do it, but also mine. This is mine. You said that this is mine, this is mine.
But what license do you want?
You have put it on the license for this.
Same license. This is my license. Yes. Let me go through that and take all the captures.
And also here, this picture is yours. When you took it, this. You have to be really careful because this has no capture. This is so that everything has a description and a capture of the person.
I understand.
This is yours. Here's the animal now there. This here, nothing. Nobody understands this. She thinks that it is good.
Yes, the net part with it.
No, I'd leave it like that. It's fine. Then make a bungee over the top. No. Good. Perfect.
Very good.
I reckon you pull the bungee on the top, you can strap that board in there. Josh called it rope, that's why.
Yes, no. This is basically.
Well, if you want, I can put bungee on it. What's that? That's it. That's it. So that rope is going here.
So the rope that it falls.
You're almost there, like you're basically there. It's just it's not position there, but good. You've got it behind the rail there. Sorry. For some reason it keeps twisting in. I don't know. That's good. Oh, I see. It's caught in that. It's like the back corner of that thing there. Keeps hitting that. See ya? This was causing you a problem.
Who makes the caption? Who writes the captions? This is the catching stuff. The flow was as follows. I convert text in a written flow. The thread I try to learn it is achieved that each tweet has an image.
Right, good.
This is good time.
Yeah. Just that's forward, that's in. Let go. Send all the way to there. It's that rope, man. It's catching every time. Right, try that. That should be down now. Yeah. You should really pull out from there.
Catching on that hose.
Nose will move. Close your jaw a little bit. Yeah, try to do it.
Can you put the rope over it? Then the other arm.
You need to get it set in first.
Yeah, it's like the yellow rope. It's over, so he was in like already several times.
The rope is catching on the
Yeah,
top,
yeah,
and
yeah.
it pulls it off.
Get in position, I'm getting the rope over it, and you pull it off.
Yes.
I can't do anything right now.
It's almost there.
The white one or no, the yellow one. Okay, like it's that's like catching up on the yeah. Oh. So he was already in like 3 times or so.
Yeah.
So.
Yeah, it's better
It's
than
caught
that.
in there.
Yeah. Okay.
If I push that corner with that head,
If I let go and grab it higher, hopefully the rope won't be able to go again.
it's catching the screws on
Yeah,
the
I see it. Yeah, yeah. But the rope was on the box, no?
Alright, so one please.
This is artificial modernized silicone sensor.
Which is if you
Hola, buenos días, buenas tardes, buenas noches, saludos de donde sea que nos estén viendo, desde donde sea que nos estén acompañando. Acá está con ustedes Ivonne Rodríguez Ramírez de la Universidad de Costa Rica a bordo de el Falcor 2 y entramos de nuevo a una sección de narración en español para que todas las personas de habla hispana puedan compartir y saber qué es lo que está pasando acá en el dive con el Sebastian. Pueden como siempre mandar sus preguntas a el chat, preguntas, comentarios o lo que quieran. Puede ser en español, en inglés, en portugués. Tenemos un crew dentro del barco muy internacional, así que cualquier idioma es bienvenido. En este momento lo que estamos viendo es imágenes de cómo están retirando una caja perteneciente a el experimento que se está instalando el día de hoy. Para recordar los experimentos que se están haciendo en este momento en las ventilas hidrotermales para poder validar la hipótesis de que microorganismos y larvas de otros animales utilizan estas ventilas como método de movilización. El experimento consta en poner una caja de malla de 32 micras encima de la ventila para básicamente atrapar todos los microorganismos y animales que estén saliendo de la misma. Para poder diferenciar cuáles organismos estuvieron presentes al momento de instalar la caja y cuáles fueron los que entraron después por medio de la ventila hidrotermal, se tiene que poner un tinte al inicio del experimento para teñir todos los organismos que estaban ahí presentes. Este tinte se tiene que quedar ahí por 3 horas, entonces para poder contenerlo en un solo lugar se tiene que poner encima de esta caja de malla una caja sólida como la que estamos viendo ahorita en pantalla. Ya se puso el tinte, ya pasaron las 3 horas, ahorita lo que estamos haciendo es recogiendo la caja y luego lo que vamos a hacer es ir a ver el arte de el artista a bordo Max Hopper-Schneider para ver cómo está, para tomar nuevas tomas. Ayer se puso la escultura de Max en una zona cercana al punto del experimento, entonces apenas terminemos de recoger la caja y de posicionarla bien en el ROV, entonces vamos a ir para allá.
Listo.
¿Sabes?
Quito la crema esto, pues.
Light's catching. It keeps heading on the elbow.
Black side.
That's better. Just hold it and get it back out.
Alright, hold on.
Acá al guardar la caja estaba siendo un poco complicado porque no cabía bien y se tiene que hacer con muchísimo cuidado, ya que no se puede romper o rayar el vidrio. Bueno, en realidad la caja no está hecha de vidrio, está hecha de un plástico muy resistente para que no se rompa al recibir algún golpe y también para poder aguantar la presión. For everyone else that is joining us right now that is not a Spanish speaker, I am Ivonne Rodriguez Ramirez from the University of Costa Rica. I am going to be speaking in Spanish for the next hour or so, but you can still send all of your questions and comments in the chat in English, in Spanish, or in the language that you feel more comfortable with.
Towards you, actually, just let it go ahead. It's not a long box.
I'll do that in a second. Look up, please.
Acá en pantalla al fondo en la parte superior podemos ver las otras dos cajas de los experimentos que ya se han instalado en los días pasados de esta expedición. El día de hoy es la tercera y última instalación de experimentos y bueno, las tres instalaciones han sido bastante exitosas, por dicha. Eran operaciones bastante delicadas que duran todo el día. El ROV bajó por ahí de las 6:30 de la mañana y se ha utilizado todo el día solo para la instalación de este experimento. Entonces, como podemos ver, es un proceso largo, un trabajo duro, pero se ha logrado hacer con éxito. En la parte derecha de la pantalla podemos ver justamente de manera muy clara cómo los gusanos Poliquetos riftia están saliendo de una de las grietas que está entre las rocas en esta zona. Justamente por esas grietas es por donde fluye el agua caliente, por donde salen posiblemente muchos nutrientes y químicos que utilizan los microorganismos que potencialmente están viajando por ese flujo de agua.
I'll have to move over to it actually because you see the signs, Chris? Yeah.
Yeah. We can touch it.
No, it's in. It just is. It's like.
I'll stop. Hold on. Bring your camera back. Bring your camera back. You might have to spin that. Gently.
Can you do it?
Gently porch in.
Can you do it?
I can't porch. I just gently porch in. Yep, I'll stop.
Right. Well, I'll have to stay there tonight to get this section. So it's right here. Yeah, that's it. Grab it, just ouch, edge here. I can't get down much further than here. That's fine. Okay, try that.
What do you do?
Pick it up.
The fish just took right
To the box.
Vimos que un pez entró a la caja del experimento, por dicha salió, así que no es, la idea no es que sea colectado. En este momento podemos ver que en efecto hay muchos peces en la zona. Estos peces son del género Termasus.
Maybe the one closest to you might be easier to grab.
La especie específica del pez es Termasia cerberus. Varias personas al ver estos peces piensan que son anguilas, pero en realidad no, son de una familia completamente distinta.
No, that's a tube.
Oh, it's a tube, okay.
It's got like this thing.
Yeah, I get it.
That's gonna be part of that. Yeah. Maybe shift the pressure.
Right, some kitchen.
Yeah, your kitchen.
Yep, I got it.
Acá justamente están guardando un equipo que habían dejado a un lado, que es una jeringa de plástico muy grande. Esas jeringas se utilizan para poner una resina, que es de las resinas que se utilizan para el mantenimiento de fondos de pisos de piscinas. Esta resina llega y se inyecta en los extremos de la grieta que quedan a los bordes de la caja del experimento, justamente para poder sellar una sección de la grieta en donde se va a poner el experimento y así poder tener mayor control de las especies que quedan atrapados en la malla de la caja. Van a poder ver que esta jeringa de plástica tiene amarrada una pesa. Justamente es para que no se vaya flotando y se pueda posar bien sobre el fondo marino. Justamente la caja de malla en la que se usa para el experimento también tiene alrededor, en la parte interna, en la parte de abajo, un montón de estas pesas negras para que se pueda posar bien sobre el fondo.
Before you get on with it, I'd like to see if you can try and notch that box. I need to get out of it.
What's that?
You can notch that box, see why it's...
I don't think that it's... Is that box 100% square?
En este momento los pilotos están chequeando que todo esté organizado bien, acomodado bien en la plataforma que se retrae del ROV. Acá en la sala de comandos hay muchas pantallas con diferentes ángulos. Acá nosotros en el live estamos viendo solo uno, pero los pilotos tienen diversos ángulos de lo que está pasando para que ellos estén seguros de que todo lo que estén viendo está asegurado, está bien amarrado, que nada esté golpeando y que todo esté funcionando bien.
So it's sitting up by about 5 millimeters, right?
Bring it towards you.
Push it here a little bit.
Bring it out a little bit.
No. No.
Gotta get clearance. So you may be nudging it.
Can you move the camera out?
Down.
Look down. Move down.
See it in the back corner there? It's not sitting up properly in the back. Probably that's causing the problem.
Push it here. The structure is there.
Tenemos una pregunta en el chat de que cuántas cajas se van a poner para completar el experimento. Se van a poner 3 cajas en total, es decir, 3 réplicas del experimento, y justamente hoy se acaba de terminar de instalar la tercera y última réplica. Estas cajas se van a quedar acá por una semana y luego con el ROV se va a hacer como un huequito en una de las paredes de las mallas y se va a succionar todos los animales que estén dentro de la caja. Luego la caja se va a recoger y también esa malla va a ser procesada, porque si ven en otras tomas de los lives van a poder ver que hay como unas manchas blancas encima de la malla, y esos son mantos de bacterias que también es parte de la muestra. Ya se pudo guardar bien la caja. Como estábamos viendo, el problema era que estaba chocando con esta pieza de metal que tiene el ROV, pero ya se pudo acomodar. Fue una tarea complicada, pero se logró. Y ahora sí estamos listos para seguir. En la parte superior derecha de la pantalla podemos ver que justo debajo del cúmulo de almejas hay varias anémonas pegadas en el sustrato.
Yes.
En este momento vamos a proceder a tomar una muestra de succión con este apéndice que tiene el ROV, que es como una aspiradora. Estas muestras de succión se procesan, se pasan por dos filtros, uno de 32 micrones y otro de 1 milímetro, para poder separar los animales un poquito más grandes de los animales más pequeños que se tienen que ver a microscopio. Todas estas muestras llegan, se guardan en alcohol y se llevarán a una universidad en Holanda para ser procesadas. Y el procesamiento de estas va a consistir en separar todos los diferentes animales, identificar todas las especies que hay, para poder tener una idea clara de las comunidades biológicas que forman parte de estos ecosistemas. También con estas muestras se está intentando atrapar copépodos, que son unos crustáceos pequeñitos, y estos copépodos son para un experimento que se está desarrollando en este momento a bordo del Falcon 2. Justamente lo que se intenta ver con los copépodos es su resiliencia. Se van a poner en diferentes cámaras presurizadas con diferentes temperaturas para poder saber si ellos podrían sobrevivir un flujo como el que se da por las ventilas hidrotermales. Otra pregunta en el chat sobre cuánto tiempo nos queda en el fondo. El ROV normalmente sube a superficie o empieza su ascenso a las 4:30 de la tarde. Así que en estos momentos nos queda 1 hora más de tiempo acá para luego empezar el ascenso. El ascenso del ROV Sebastian también es pasado por el livestream y les recomiendo que lo vean porque siempre se ve mucha fauna que vive en la columna de agua. Muchos tenóforos, medusas, sifonóforos y todos estos animales gelatinosos que tienen bioluminiscencia, iridiscencia y son muy bonitos. Si se preguntan cuál es la dinámica después de que el ROV sube a superficie, lo primero que se hace es asegurar de nuevo el ROV en la plataforma especial que tiene a bordo del Falcon 2. En este momento el equipo científico se prepara con baldes de agua fría para poder colectar todas las muestras. Y luego las muestras se separan en los diferentes equipos dentro del grupo científico para los diferentes experimentos. El equipo científico termina trabajando y procesando sus muestras y poniendo sus experimentos a funcionar durante toda la noche. Entonces es un trabajo básicamente de 24 horas que se tiene que hacer en equipo, de un equipo hecho por el crew del Falcon 2 y el equipo científico.
Okay, this is an example. This is right there.
No, it's the...
start.
Si le ponemos atención al agua, podemos ver que hay muchos puntitos blancos y como partículas suspendidas moviéndose en ella. Muchos de esos puntitos son también animales pequeños, entonces justamente esos son los que queremos atrapar con este apéndice de succión que tiene el ROV. Hay una pregunta en el chat sobre la especie de pez que se ve acá en el fondo. La especie de este pez es Thermaces cerberus.
So we got all the way around.
Estos peces son bastante abundantes, es la única especie de pez que hemos visto hasta el momento en estos sitios y de hecho es de los organismos que no se están colectando y con los que no se está trabajando ahorita a bordo. La expedición se está enfocando más que todo en los gusanos poliquetos Riftia, que vemos acá, estos de tubos blancos grandes con plumas rojas muy prominentes. También nos estamos centrando en las almejas que están asociadas a estos sitios que podemos ver normalmente mezcladas con los gusanos. También se está trabajando con las rocas del fondo, con los dos animales pequeños que estén flotando cerca, digamos, del fondo y dentro de las comunidades que estamos colectando en este momento. Y esa es la fauna en la que se está centrando la expedición hasta el momento. Las rocas claramente no son fauna, pero estas se colectan porque hay una estudiante a bordo que está investigando las comunidades de bacterias que viven dentro de los poros de las rocas.
Bring down the bacteria and viruses from the ship to the...
If there's a way to not do that.
Yeah, first of all, the viruses they would not survive because they adapted to live like in the air or on the surfaces here on the ship and not in the sea.
Right, Jeremy, that's off seabed. We're going to start commence covering.
And other than that, we need to disinfect the RV. But so I'm not concerned about viruses and bacteria, but what you do have to be careful, what you do is that it's not transplant organisms, specimens. Because you can usually like check very easily.
Acá, tenemos una pregunta en el chat preguntando que si existen mesuras para evitar la propagación o introducción de virus y bacterias ajenas a el fondo a través del ROV. Le hicimos la consulta a una de las investigadoras principales, la doctora Sabine, y ella nos estaba contando que en realidad no hay ningún protocolo específico para esterilizar el ROV antes de bajar al fondo, pero que eso a ella no le preocupa porque las bacterias y viruses que viven en la superficie están más acopladas a las características de la superficie y no a las de los ecosistemas del fondo. Pero con lo que sí hay que tener cuidado es más con la posible, digamos, introducción de especies distintas que no sean virus o bacterias al fondo. Entonces, en conclusión, en realidad no hay ningún protocolo de esterilización o de evitar que haya una introducción de virus o bacterias al fondo. En caso de que hayan personas nuevas que apenas se estén conectando, saludos, acá les habla Ivonne Rodríguez de la Universidad de Costa Rica y en este momento están viendo el live stream de la expedición al fondo del Falcor 2 por medio del ROV Sebastian. Bueno, ya en este momento nos estamos alejando del fondo. El espacio en español, la aproximada hora acaba de terminar. Muchas gracias por acompañarnos. Ojalá que puedan ver todo lo lindo que tiene la columna de agua en este momento. Muchas gracias y ojalá nos puedan acompañar mañana. Recuerden que cada día alrededor de las 2 de la tarde, hoy se atrasó un poco, pero bueno, pero alrededor de las 2 de la tarde vamos a tener siempre un espacio en español para toda la comunidad hispanohablante, así que ahí nos vemos.
All right, well, thank you so much to Yvonne for sitting in. We have left bottom, so we're currently going to be ascending up through the water. I'll see if I can get some final words from our scientists here momentarily, but they're kind of conferring with each other, I think, for our future plans. Today seemed like a pretty good day. We've got third experiment in place, and we were also able to go around and show some of the science that's going on behind the scenes on Falcor 2. Obviously, the dye box is just what we're seeing on stream, and the vents, the obvious parts of the vents we can see as well. So being able to share just the unique and often very surprising and interesting work that's being done on the ship was very fun today. So I hope everyone appreciated that. And we'll be leaving the camera on for Ascent, but let's see.
Good. So again, we had a very nice dive today. We could successfully deploy our experiment number 3. So yesterday we had looked for the area, and today in the morning we cleared the area a bit more with the suction sampler. Then we sealed the cracks with the glue, put the mesh box on top, put the staining box on top, added the dye to let the animals stain, waited for quite some time, and afterwards removed the dye again, put the staining box back to the rough, and now we are on our way up.
All right, that was a great little recap of the dive. So as you heard, we'll be leaving the camera on for the Ascent. For those of you who have never been around for the Ascents on these, it's going to be a lot of looking at water for sure, as we have over 2 kilometers to ascend back to Falkor 2 for recovery, but you never quite know what you're going to find in the midwater. Yesterday was pretty fun. We had some cool stuff, and so we shall see what we find today, but the mics will be going pretty quiet for the most part. We might hop in every now and then, but enjoy the Ascent. Thank you for joining us on the dive today, and we look forward to you joining us again in the future. For folks who haven't been on any of the ascents after these experiments, we do disperse the dye on the way up. This dye can't be reused now that it's been used for this experiment, and we would hate to have the bag kind of just burst in the hangar of Falcor 2, or otherwise figure out how to get rid of a messy stain like that. So you might see it kind of creeping up from the bottom of the screen. We're not losing anything important. Sebastian's, not losing any weird fluids or anything. It's just the dye being dispersed. All right, with that, we have our floats all off, and we are going to be recovering ROV Sebastian. So I'll leave the camera on as we recover, but thank you all for joining us for another pretty fantastic dive today. We achieved a lot with the placement of that third experiment, and I hope you all enjoyed learning about some of the science going on more behind the scenes on the ship. So we'll see you tomorrow for another dive back down to the vents.
Definitely in person.